Yeah.
I mean, we senior, we do, but young people, you know, they don't. And now, you know, we are in the state of calamity. We are in the state of calamity in Europe, okay? So yesterday, our prime minister just announced more restrictions. Of course, we don't make lockdown because lockdown is going to affect economy, but restriction we need to do. So using mask in the street is mandatory now. Yeah. You see, we have a very good gift from our neighbors, Spain. They sent us many Erasmus students.
Good.
But the student brought us COVID.
Good.
Sayın Rektörüm hoş geldiniz. Yusuf Hocam.
Merhabalar Nazım Hocam, günaydınlar.
Günaydın, nasılsınız, ne var ne yok?
Gayet iyiyim. Siz nasılsınız?
Vallahi hocam yani bu online çalışma yani sabahlar şimdi YouTube'dan canlı yayın veriyoruz hocam. Onun ayarlarını yapıyorum. Bizim teknik eleman da hocam korona olmuş. Öyle olunca bütün şey güzel.
Merhabalar Nazım Hocam, günaydınlar. Günaydın. Gayet iyiyim. Siz nasılsınız?
Vallahi hocam yani online sabahlar şimdi YouTube'dan canlı yayın veriyoruz hocam. Bizim teknik eleman da hocam öyle olunca ben şey mi duyuyorum tekrar bu.
Nazım, could you ask all participants mute their microphone
Yes,
when they are silent?
Yes hocam, not all. Yes. Başladı. Bu şey YouTube şeyini nasıl oldu? Ben şeyi görüyorum burada da o sıkıntı. Ben YouTube'u burada duyuyorum. YouTube'u duyuyorum. YouTube'a yazdım şu anda da. Yani artık şu anda çok fazla bir şeyim yok. Yani YouTube. Yani. Hocam sen şey yap.
Good morning, dear guests, dear participants, dear international organizing and scientific committees, and dear sponsors and supporters of the 6th International Mediterranean Symposium on Medicinal and Aromatic Plants. On behalf of the organizing committee of the MESMAP Symposium, I would like to welcome to all our guests and participants, I would like to thank their valuable contributions to these scientific events also. Coming from more than 20 countries, about 150 participants from different disciplines such as agriculture, biology, chemistry, medicine, pharmacy, forestry, biotechnology, pharmacognosy, aromatherapy, and other research areas will discuss developments in medicinal and aromatic plants and share their experiences and with each other during this meeting. MESMAP Symposium was organized by academicians and researchers from distinguished world universities, research institutions, non-governmental organizations, and agencies. And main organizers and sponsors, you can see the list of the main organizers, sponsors, and supporters of the MESMAP Symposium in this slide. After the symposium, abstracts of the presented as oral and poster studies will be published in MESMAP abstract book with ISBN number as online abstract book PDF format. If no presentation is given by the participant, the accepted study will be removed from the online abstract book. And also, the authors can send the full text. It is not mandatory. They send the full text and it will be published our 6 MESMAP proceeding books with ISBN number as only an online PDF format. In this slide, you can see the contracted journals. Also, our participants have opportunities to publish their full papers in the contracted journals. Full papers of the studies can also submit. We have 4 journals. You can see the names of the journals in this slide. And we will also inform the participants about the publication of full papers after the symposium. Molecules have Molecules journal give us the opportunity to publish full papers in that journal, and other journals is not indexed. Annals of Phytomedicine is emerging science station index. Current perspective for medicine and aromatic plants is our journal. Professor Dr. NAZIM SEKEROGLU is the editor of this journal. You can send your papers to this journal also. And we are honored to announce that MESMAP Symposium are indexed of Conference Web of Science Proceedings Station Index Science Scopus Index. Also, this meeting has been supported by TÜBİTAK, the Scientific and Technological Research Council of Turkey, National Scientific Grant Program. We are honored to announce that we happy. And the program, I would like to mention the general program of the symposium. The first day, today, all the presentations will be on the at the Salon A, and you can use the same ID and passcode for connection to all salons and all the sessions. You can use the same ID. Today, only invited speakers give us their invited lectures, and tomorrow, the second day of the symposium, our speakers attending our congress with oral and poster share their research to us, and the presentations will continue in both salons, Salon A and Salon B, simultaneously. And poster presentations will be tomorrow. We will release the posters as online PDF format from our website, and also we will announce the poster presentations our YouTube channel also. Session chairs are responsible for the presentation and scientific discussion of the session, and attendance certificate will be sent to the email addresses of the participants as PDF format after the symposium. Despite the coronavirus outbreak, we have participants and speakers almost all over the world, and we would like to thank our valuable participants who didn't leave us alone despite the coronavirus pandemic. Invited speakers and participants from different countries. And invited speaker list you can see on slide. Professor Rao from USA, Professor Randolph Arrow from United Kingdom, Professor Yusuf Aran from Turkey, Professor Ivan from Slovakia, Professor Çimen Karasu from Turkey, Professor Jambuk Seo from China, Professor Ramandan from India, Professor Monica Hangianu from Romania, Professor Takashi Watanabe from Japan, Professor Marina Pino from Romania, Professor Alban Ibraliu from Albania, Professor Eldar Garea from Azerbaijan, Professor Mutlu Aytemir from Turkey, Professor Salvatore Labella from Italy, Professor Ali Bilgili from Turkey, Professor Berat Özçelik from Turkey, Professor Havan from Thailand, Professor Nazlı from Turkey, Professor Kuntal Das from India, Professor Yavaret from Thailand, and Dr. Hariprasad Devkota from Japan. And now I would like to invite Professor Dr. NAZIM SEKEROGLU, President of the MESMAP International Symposium, to give us his welcome speech. Thanks for your attendance and a very fruitful meeting with a taste of science. Thank you again.
I would like to thank you, Dr. Sevgi. Now she is associate professor, very new, and I would like to say welcome to our online session, opening ceremony of MESMAP Symposium. So I want to say good afternoon, have a nice day, good morning, very early in some places, and this time we are very happy to receive many participants all over the world, from Japan to Peru, from Canada to Saudi Arabia, from Romania to Algeria, Portuguese to Thailand, and many more countries. So this encouraged us to organize another meetings, and so we know that our symposium, MESMAP Symposium, reach a very good level, scientific level. Also, Sevgi mentioned that our symposium indexed by the Web of Science Conference Index. So we started the first MESMAP in Cyprus, Northern Turkish Republic of Northern Cyprus, and then organized in Turkey and Cyprus again. This is the 6th edition. Normally, we plan to organize the symposium in Izmir, Ephesus, the antique city of Turkey, a very good place and a very good hotel. But COVID pandemic stopped us, and we wait some more. And in October, also we tried the hybrid congress, but the last minute precautions from our ministry, we couldn't manage hybrid, and then now we are here online. But we are very happy to see all over the world and all of our friends. And we say in Turkish hocam, in Thai people say ajan. And evet, biz burada çok sayıda hocamız ve arkadaşlarımızla buradayız. Evet, yes, we can see the pictures, other symposiums. Cappadocia was a great symposium. I hope so. We will arrange next feature symposium face to face, and all we are praying, and we wish to all our friends happy and healthy life, and we will meet you very soon. We believe, and we will try. I hope so. Our scientists will find the vaccine and medicine, and also we believe that the source is medicinal plant, the main source and sustainable source. We should look for medicine, vaccine from medicinal plants. I hope so. We will find. We believe our professor, our scientists, and we entertain here from many disciplines, scientists all over the world. And I want to give the order, Professor Anek, short opening speech. So he is very valuable and support us for Mathematics Symposium recently. And again, I wish a fruitful Congress, and thank you very much for your kind participation. We will attend 2 days symposium, and now I give my order to Professor Anek. Thank you very much. Professor Anek. Professor Anek, can you hear me?
Okay, it's now. Okay. Good morning, Professor NAZIM. Good morning, Sepchi. Good morning, Professor Phyllis. Good morning, guests. Good morning, all participants. I will be very brief because it is very early here. I would like to welcome you for Mathematics sake. It is very unfortunate indeed that we have to do it online, because we have planned to have it in Izmir, a wonderful city in Turkey. But anyhow, we are here together to share our knowledge, to see each other, and everybody is well. And I hope this crisis is gone very soon and we can see each other again. I hope that this 2 days will be enjoyable day. I know that conferences online is not very easy. It's very different from in person, but I hope all of us will keep on and listening to many new information during this very difficult year, and good luck for everybody. Thank you.
Dear guests and participants, now we are beginning the first session, and the first session, Professor Can Bokseo and Professor Mutlu Aytemir are the session chairs, and Professor Yusuf Baran is the first invited lecturer in the session. Professor Can Bokseo and Professor Mutlu Aytemir.
Thank you. I'm here. Yes.
Yeah, okay.
Can you hear? Yes.
Thank you. Dear participant, I also would like to welcome to you MESMA 22. Even though we are not able to get together as planned due to these extraordinary pandemic conditions, I hope that it will be normalized until the next meeting. It's an honor for me to be a chair of the section 1 with Şembo Şao, and with your permission, I would like to introduce to you first speaker, Professor Dr. Yusuf Baran. He is a rector of Izmir Institute of Technology and a lecturer at Department of Molecular Biology and Genetics in Faculty of Science. His presentation is titled as Mechanism of the Multidrug Resistance and its Reversal in Hematological Malignancies. Please, Dr. Baran, the microphone is yours.
Thank you for this very kind introduction. So I want to, first of all, I want to thanks to Professor Dr. NAZIM SEKEROGLU and Professor Dr. Sevgi Gezici for their kind invitation to give a lecture in this international meeting. And also I want to say good morning, good afternoon, good evening to participants from around the world. And it's a real pleasure for me to be here with you. And Professor SEKEROGLU knows that he invited me to join this meeting a few times before, but unfortunately I couldn't make it. But this time I just, dedicated myself that I should be there and I should give my speech. So right now I'm in Ankara, in the capital city of Turkey, in a hotel, but I'm prepared to give this conference. So can you please let me know if you could see my screen now? So everything is all right?
Yes,
Yeah,
we can see.
Yes.
Yes, okay.
Yes, we
Perfect.
can.
So I'm going to talk about the molecular mechanisms of multi-drug resistance and its reversal in hematological malignancies. In this figure, you can see, we bring the patient from around the world together and then we name them with a disease. And it can be breast cancer or chronic myeloid leukemia or brain cancer. And then after we name the disease, then we start to treat the patients with different approaches. But the response of the patient or the response of the population is different from each other. Sometimes you can get a very good response from the treatment and the patients get cured. But sometimes, unfortunately, we don't get a response from the patient. Or even sometimes there is an intolerance, which means the patients cannot tolerate the applied anti-cancer treatment protocols. The main reason, while the disease is the same, while the doctor is the same and the drug is the same and the treatment protocol is the same, but the response is different from one patient to another. And of course, this is something regarding with our genetic background because our genetic is different from each other. So what happens in the research labs, what we see in our lab? Here you can see we get a cancer cell line and then we start to treat them with a drug. And then 90% of the cells are undergoing apoptosis, while a small percent of the cells can survive and maintain. Then they can start to grow and proliferate. And then we can go the second round and then the third and then the fourth. And after the end of a time, then we can develop drug-resistant cell lines. And then we can start to compare them with the parental sensitive cell line. And then we can see the differences between the genetic backgrounds. Well, there are many different mechanisms that are involved in drug resistance. And the number one is regarding with the accumulation of the drug within the cell, because the drug should get into the cell to become effective, to become apoptotic agent. But normally in our genome, we have like 40 genes involved in human ATP binding cassette transporter genes. And normally they are expressed on the cell membrane, and their main function is to export the artificials from the cells to the outside of the cell, or when there is an chemical agent trying to enter inside of the cell, they do not allow the toxic chemicals to get into the cell, and they export the toxic chemicals produced within the cells. So normally, these ATP binding cassette transporter genes are there to protect our cells. But when these genes are overexpressed on cancer cell lines, then your drug, which should be effective to get into the cell, cannot get into the cell. And then, this is one of the mechanisms of the drug resistance. From the left bottom side, you can see the 3D structure of these MDR1, MRP1, and BCRP, and they are mainly involved in export of the drugs to outside of the cell. And in our studies, since my time is just 20 minutes, I will be quite sure to talk about all the mechanisms involved in drug resistance. In our studies, we have observed that the acute myeloid leukemia cells resistant to vincristine and doxorubicin overexpressed MDR1 and MRP1 to export the drug to outside of the cell. And when we inhibit these transporters with different chemotherapeutic agents, then we observed increased efficiency or treatment efficiency of the drug on the cancer cell lines. As you know, we are now very good with proteomics and the computational biology as well. And within the cell, we can very easily define the 3D structure of the proteins, which can be the target for the cancer treatment. And then we can design specific anti-cancer agents that are specifically recognized and binds and inhibits the cancer-inducing proteins, metabolites, lipids, whatever. So the main mechanism is like a key and chain, you know. So the specific treatment agent comes and binds to the cancer-inducing initiating protein and then inhibit it. But since the proteins are translated from the mRNA, which is transcribed from the DNA, any mutation in anti-cancer agent recognition site can decrease the efficiency of drug binding and, of course, efficiency. Here you can see a reciprocal translocation comes together between long arms of chromosome 9 and 22nd, which brings ABL and BCR gene together, and then they form a strong conversion of a proto-oncogene to an oncogene. And here you see the signaling pathways regulated by BCR and ABL. Through different genes, activating of different genes, BCR, ABL induce growth and proliferation and inhibit apoptosis. And then there was a drug developed in 2000, which is imatinib. Here you can see the action mechanisms of BCR-ABL. There is a substrate binding region, an ATP binding region. When ATP and substrate bind to BCR-ABL, then BCR-ABL moves one of the phosphate from ATP to substrate, which induces the homogenesis. But if we could develop a drug that competes with the ATP to bind the ATP binding side of BCR-ABL, then we inhibit the phosphorylation of the substrates, then activation of the leucohomogenesis. But while the imatinib specifically recognized 21 amino acid in BCR-ABL ATP binding site, when there is a mutation in any one of these amino acids, then the binding efficiency of imatinib is going down. The same is true for nilotinib and dasatinib, which are the second tyrosine kinase inhibitor. And then, of course, there is a third line TKI inhibitors as well. But at the end, when there is a mutation on ATP binding cassette side of the BCR-ABL, then the drug cannot bind and cannot inhibit the activity of these oncogene. And the third one is the increases in drug targets. While you inhibit any protein within the cell to treat the cancer cell and to induce apoptosis, but of course, you should be between the minimum effective dose and maximum tolerable dose. You cannot say that, okay, just increase the amount of the drug that you deliver, but there is a maximum tolerable dose. And your target is protein. Here you can see the signaling pathway regulated by BCR-ABL. And here you can see the cell line that we developed for imatinib resistance, which is 1 micromolar imatinib resistant cell line and 0.2 micromolar imatinib resistant cell line. And on the bottom of the figure, you can see IMA 1 micromolar resistant cell line is like 20 times more resistant to parental sensitive cell line. And here we checked the protein levels of the BCR-ABL, because in these cancer and drug resistant cancer cell lines, we are targeting BCR-ABL. And here you can see on the left upper part of the figure, it's a Western blot analysis of BCR-ABL. Here you can see when the cells become more resistant, then the protein levels of the BCR-ABL is going up. And the same is true for the overexpression of BCR-ABL as well. So here you can see when the cells become more and more resistant to imatinib, BCR-ABL expression and overexpression is going up. The same is true for the right side of the figure. Here you can see MEC01 cell line, and there is an increase in protein and mRNA levels of the BCR-ABL. It is not only an overexpression or the translation, but in this figure, we check the protein stability in resistant and the sensitive cell lines. So we inhibited the translation machinery with cycloheximide, and then we check the protein levels of the BCR-ABL in sensitive and resistant cell line. And here you can see after 24 hours, and with different time intervals, here you can see 3, 6, 8, 10, 12, 24, 30, and 36 hours. Here you can see after 24 hours, there is a significant decrease in BCR-ABL protein levels in sensitive cells. And after 30 and 36 hours, there is no BCR-ABL protein. But for the resistant side, there is protein, BCR-ABL protein, even after 36 hours, which means resistant cell lines increase the stability of the BCR-ABL protein as well. The main aim in cancer treatment is the induction of apoptosis. And here you can see very briefly how the apoptosis signaling is regulated within the cell. While on the upstream side, you target the induction of apoptosis between the midstream or the downstream side of the signaling pathway. If somehow a mutation, a deletion, a downregulation, or even upregulation of certain genes happen, then no way to induce apoptosis. So you know MCF7 cell line, which is caspase 3 deleted cell line, which means you cannot induce caspase dependent apoptosis in MCF7 breast cancer cell line. And here what we observed on the left side of the figure, you can see after 500 nanomolar imatinib application, 80% of sensitive cell population was in apoptosis, while there was no mutation, while there was no apoptosis in drug-resistant cell line. The same result was happening for MEG01 cell line, which means drug-resistant cell line shows resistance to apoptosis. But what happens over there? And here we checked the apoptotic and anti-apoptotic gene expression. And here you can see in resistant cell line, there was an overexpression of BCL2 and XL, while a downregulation of BUX, which is a pro-apoptotic gene. So through the change in expression levels of these apoptotic and anti-apoptotic genes, apoptosis was inhibited. So dear participants, ceramides, bioactive sphingolipids, lipids are known to be involved in cell growth and proliferation. They were known with their structural functions by 20 years, but now we know that they are involved in cell growth, proliferation, cancer, apoptosis, drug resistance. And here you can see a very general signaling pathway of the bioactive sphingolipids. On the upper side of the metabolism, you can see de novo synthesis of ceramide. And the ceramide is the central molecule of this pathway, which is very strong apoptotic molecule. But when the ceramide was converted into sphingosine 1-phosphate by SK1 or converted to glucosylceramide by GCS, then this very strong apoptotic molecule is converted to a very strong anti-apoptotic drug-resistant inducing molecule. And here you can see how ceramides are activating their downstream targets, which are telomeres, telomerase, c-MYC, caspases, cyclin-dependent kinases, mitochondrial membrane potential, and at the end, they induce apoptosis. In this figure, we treated the sensitive and the parental cell lines with 500 nanomolar of imatinib and then checked the intracellular concentrations of the ceramide. And as you can see for C18 ceramide, imatinib application significantly induced overexpression of SIRS1 gene, and SIRS1 is responsible for C18 ceramide generation. When you compare column 1 and 3, you can see in sensitive versus resistant cell line, there was a downregulation of ceramides. And then after observing and having these results and many more, then we focused on the yellow side of the lipids, which is conversion of ceramide into sphingosine 1-phosphate through sphingosine kinase 1. So what happens with this sphingosine kinase? When there is a growth factor, it's phosphorylated and activated. Then it converts sphingosine to sphingosine 1-phosphate. Then S1P goes outside of the cell, binds one of these 5 receptors, then activate this signaling pathway and induce migration and survival, proliferation, and inhibit apoptosis. So what happened in our result? What happened in our studies? We observed that SK1 enzyme activity is going down in sensitive cells in response to imatinib, while it's going up in resistant cells, and in response to drug application, even going up. On the right side of the figure, you can see S1P levels showed the parallel result with the enzyme activity because it's the product of the enzyme. And on the left downside of the figure, you can see downregulation of SK1 increased the apoptosis, while upregulation of SK1 showed increased resistance to imatinib. So we check the both sides of the coin. When SK1 is upregulated, then apoptosis is going down. When SK1 is downregulated, then apoptosis is going up in response to imatinib. And here you can see in more resistant cell line, there was an overexpression of SK1, both in mRNA and the protein levels. By that time, BCR-ABL was known as the top of the pyramid for the initiation and induction of chronic myeloid leukemia in hematological malignancies. And there was no gene, there was no mechanism identified that regulates the expression levels of the BCR-ABL. And we were the #1 showing that there is a gene on the upstream part of the BCR-ABL regulating its expression. And for that one, we upregulated SK1 through plasmid vector induction, cloning of the SK1. And then we see that there was an upregulation of BCR-ABL as well. And when we downregulated SK1, then BCR-ABL downregulated as well. So we also checked both sides of the coin and observed that SK1 regulates BCR-ABL expression, but through which receptor? Then we downregulated all the receptors, S1P receptor 1, 2, 3, and 4, and with the scrambled siRNA. Then we checked that, we observed that receptor 2 is involved in SK1 regulation of the BCR-ABL. And then on the right side of the figure, you can see when there is a 315 amino acid mutation in BCR-ABL side of the ATP binding side of the protein, there is no way to treat this patient with any kind of chemotherapeutic agent. You should go back to stem cell transplantation. Here you can see nilotinib is one of the most effective drugs in CML treatment. And you can check the BCR-ABL expression levels in response to control. So nilotinib application could only decrease BCR-ABL expression for 25%, while SK1 inhibitor was around 60%, while receptor 2 inhibitor, which is JTE, is around 90%. But when we combine SK1 together with receptor 2 inhibitors, then there was like 98% decrease in BCR-ABL expression levels. Then we checked the same result with the patient samples who are in the blast crisis. And on the fourth column, you can see SK1 and receptor inhibitors significantly decrease cell survival in different cell line. So then we checked the green side of the sphingolipid metabolism, and we observed that there was an overexpression of GCS in imatinib-resistant cell line, but when we inhibit GCS with PDMP, we observed significant increases in apoptosis in response to imatinib in sensitive and resistant cell line. Here you can see that there was no apoptosis in response to imatinib, but when we downregulated GCS with PDMP application, you can clearly see an increase in apoptotic cell population here is as well. So all these data were coming from the cell line studies. Then we moved to real patient samples involved, the bone marrow samples. And here you can see the patient number 1, 3, and 5 are newly diagnosed. The patient number 2 is positive response. And the patient number 4 is drug-resistant patient. And here you can see in patient showing positive response, there was an overexpression of SIRS1, SIRS2, SIRS5, here you can see SIRS6. And when we come to the GCS in drug-resistant patients.
Dr. Baron, last 2 minutes, please.
Okay, I am okay with that, and I am happy with that because I'm almost done. Here you can see the GCS expression is going up in drug-resistant cell line. So when we get the patient samples from the hematologist, and after checking these gene expression, they are the genetic signatures, we clearly foreseen which patient can show resistance can show the sensitivity to drug applied. And here you can see more valuable result from the patient samples. Here you can see the same patient with different time points. So when the patient treated with TKI and when they showed positive response, here you can see the GCS is going down SK1 is going down and BCR-ABL is going down. But newly diagnosed versus drug-resistant cell line, GCS is going up, SK1 no change. So all these analyses showed us that bioactive sphingolipids can be used as a diagnostic, prognostic, and targeting agent for the treatment of the chronic myeloid leukemia. But of course, increases in DNA repair, epigenetic differences, involvement of microRNAs in the regulation of the genes involved in drug-sensitive resistance, apoptosis, growth, and proliferation. They all are involved in drug resistance and sensitivity as well. But just because of the time limit, I will stop here because there are like 400 articles, abstracts in national, international meetings behind these 20 minutes talk. So I just try to be as short as possible. But at the end, I can tell you that each patient's cancer is as unique as the human fingerprint. And we have to know about the genetic differences, genetic signatures, and then treat the patient according to their genetic background and according to their responses. There are many different funding agencies, including European Union, Turkish Hematology, Turkish Academy of Sciences, the NSF of Turkey, Izmir Institute of Technology. Once again, I want to thank a lot to Professor Dr. NAZIM SEKEROGLU and Professor Dr. Sevgi Gezici for their kind invitation and giving me the opportunity to share the data that we generated in my lab. And also, I want to thank you all for your being here and for your patience. Thank you so much for being here.
Thanks you your very inspiration presentation, Dr. Baran. And by the way, we are also working on the same subject as yours. I would like to have future collaboration with you if it's convenient for you. And also we have get only one question or two questions.
Sure. I'll be more than happy to collaborate, and this opportunity is open to all the participant scientists from around the world. As the president director of Izmir Institute of Technology, and we are ranking #1 for articles per faculty, and we are ranking #3 in general in Turkey. So we will be more than happy to collaborate with your universities, with your departments and labs. I'm quite open to international collaborations. And just one more data, 45% of the articles published from my university is with international collaborations. So we are quite an international university and we'll be happy to collaborate with your labs and institutes.
Is there any question? No. Okay. Shambo Shao.
Yeah, I'm here. Yes. Yes, I want to invite the next speaker, Professor Randy Arrow. I think he's my friend. We met many years ago. He is good at his research is on the dietary flavonoids. Today we will talk about the absorption, distribution, metabolism, and excretion of dietary flavonoids and their glycosides. Randy, you can start.
Okay, thank you very much, Shambo, and I'd like to thank the organizers, Professor SEKEROGLU and all the other organizers for giving me the opportunity to share my thoughts with you at this meeting. I mean, it's a pity we can't come to Izmir, but things are as it is, and I have to congratulate you with getting a meeting together at all. So I'll see if I can share my screen now. Yeah. Almost there. So you can see this. Okay.
Yes, we can see. Yeah.
Okay, thank you.
Perfect.
Okay. I work at the School of Pharmacy in Leicester, where it's where it's just early morning now. I can now just switch off the light. The sun has come up now. And Leicester is a town in Middle England. It lies, you see here, England, Wales, Scotland, Northern Ireland, the parts that make up the United Kingdom. And you see London is quite south. People often think that London is here in the middle, but the town there in the middle, that is Leicester, in the middle of England. And I'm quite interested in many products, not so much immediately medicinal products, but products that are in our diet and still contribute to our health, so have some kind of medicinal figure. And in this sense, I'd like to cite someone from not far from Izmir, Hippocrates of Kos, an island just off the Turkish coast, known as the father of medicine. And he already stated a few millennia ago, let food be your medicine, and wrote extensively about that, or in his works called the Corpus Hippocraticum. And he had a point, because of course the Mediterranean diet is now widely recognized as good, not only for the nutritional purposes, but also for health purposes. And the question is, what is in it? What is in the diet that makes it so attractive and makes it it seems to help in the prevention of cancer. It seems to help in the prevention of arteriosclerosis and a whole range of inflammations, as reported in the journals that we are all familiar with: Planta Medica, Ethnopharmacology, Phytotherapy Research, all the journals that are dedicated to natural products analysis. So there are plenty of it, and numerous papers are written on the topic of ingredients in the diet that have an impact on our health. But it's not, there are also epidemiological studies. I just mentioned one, but there's a new one pretty much each year, confirming that the diet plays an important role in keeping us healthy. The World Cancer Research Fund has an annual update, which you can find here on their website, and it's not easy to find out what it is in the diet that contributes particularly to our health. But there are 6 reports now. Well, I say 6 reports. It's now continuously updated. So after the 7th report, there's just continuous update online. But there are some recommendations that they are clear of. They are normally very cautious in their predictions, but limiting red meat seems to be so. All the things here in red should be avoided. Red meat, grilled meat, poached, grilled meat, anything that is slightly burned should be avoided. And the total fat intake should be limited. Salt and salty food should be limited, but particularly a diet rich in vegetables should be promoted. They mention, of course, vitamins and antioxidants. I'll get back on this whole antioxidant idea in a moment. But also particularly mentioned are phytoestrogens in pulses, in soybeans particularly, that have they kind of mimic the function of human hormones, and they can play a role through that. And the evidence is mostly from case control studies, but so the evidence is not very strong. It tends to go to the health direction, and it's not easy to investigate. So some of the work that we've done in Leicester is on the phytoestrogens, and here you see that they have a kind of rough structural similarity with estradiol, the normal estrogen hormone, and you can map them on top of each other. So you can kind of imagine that variations of this molecule will fit into the same receptor sites as the estradiol does. It has the hydroxyl group here. It has, these all have a hydroxyl group here too. It has the other one here. So there's hydrophilic places, there's hydrophobic places, and the overall shape and the distance of the different groups make that they would, if you take the lock and key principle, they would fit in the same lock, but some better and some worse. And over the years, long lists have been made of particularly flavones with different substituents on the rings. You see here the groups are 1, 2, 3, 4, 5, 6, 7, 8, 9. And you can have all kinds of variations there. Mostly there are OH groups, hydroxyl groups, but they can be methylated, so you have methyl ethers, and you can have prenyl groups attached and sugars. So there's a whole range of them, and they all act as estrogen analogs. It means so that they have an interaction with the estrogen receptor, either blocking it or stimulating it. And based on that observation, people have done in vitro experiments with cell cultures, and they come up with these nice pictures of cell signaling diagram, similar to the ones that we saw in Professor Barron's presentation just before this one. This work is very important because it tells us how molecules can work and how molecules that are in the diets, or medicinal molecules for that matter, interact with the cell and exactly how the mechanisms work. And it can be shown that the flavones in particular have an action in controlling factors that affect inflammation. So COX-2, INOS, TNF-alpha, IL-6 and nitrous oxide, they can all be, the amounts can all be reduced. It's often mentioned, particularly for the flavones and the polyhydroxy phenols, that they have antioxidant activity. And then often that is supported by some tests where a reagent is mixed with any of these flavones, and you can see that oxidation is prevented. And then the story tends to be, so if we eat these antioxidants, they are in our body and they circulate here and they do their antioxidant effect there. I'm quite critical of those type of publications, because the amounts that you would need to get the antioxidant effect that is measured in vitro, in test systems, the amounts are too high. They are not necessarily circulating in the body. But what is possible is that small amounts of the molecules of this type that you see circulating in the body can have an effect on receptors or hormone receptors, possibly, and then kind of start an anti-inflammatory response. That is a model that I can understand. So to come back to Hippocrates and his idea that let food be your medicine, there are a few things that are obvious. Food, of course, provides us with nutrients that we need to still our hunger and to grow and to actually be able to live. So the nutrients are in food, and then there are some constituents that are essential in our diet, the essential amino acids, of course, essential fatty acids, but also things like vitamins. So we all need to have them from our food. But all the other effects, like the antioxidant effect and the triggering of antioxidant enzymes, are caused by this third group of compounds, so nutrients, vitamin-like compounds, and then we have what is now often referred to as phytonutrients. So here are a number of them, the flavones and flavonoids, They all are derivatives of this naringenin, and you have to imagine all these molecules with various substituents on the ring, often polyhydroxy type compounds, and they are part of our diet. And there's even a structure-activity relationship of the molecules. As I look at my watch now, so I go a bit faster. So we know that these molecules can do something in vitro, but what do they do in vivo, in real people? Thing is that many of these compounds are not easily absorbed in the human gut, and they don't occur as they are tested as aglycons, as the molecules themselves, but they're typically linked with sugars. So they're glycosides, the molecule linked with a sugar. And the sugar can be glucose, then we call it a glucoside. It can be another type of sugar, like a rutinoside here, and then it's a rutinoside. So there are many different types of glycosides, but I'd like to make a distinction between the glucosides with the glucose and other ones, because once they are in our intestines, and this is a diagram showing the cells in the lining of our gut. This is the inside of your intestines, and here at the bottom you see the bloodstream. And of course, if you want these molecules to act in the body, they need to enter the bloodstream and then be transported to the parts of the body where they are needed. And the fact is that glucosides with glucose attached to them are quite easily absorbed. There are systems here that can either clip off the glucose molecule, and then the aglycon can freely move into the cells and then be further modified and then move to the bloodstream. Non-glucoside glycosides are not absorbed because we don't have the enzymes to break off the sugar, and we don't have the receptors to pull the whole glycoside into these cells and then transport it to the bloodstream. So only a part of the molecules are actually absorbed directly. And once they are absorbed, they are often metabolized, for example, by catechol or methyl transferase. So here we have a typical polyhydroxy molecule, and you can see methyl groups can be added either here or there. There are different methyl transferases circulating in the body. So the molecules that are tested in the lab are not necessarily the molecules that arrive at the point of action. And there are further conversions in the body referred to as type 2 detoxification, where glucuronic acid is added to the molecule for sulfate groups, and the glucuronic acid and sulfates can also be added to the already methylated compounds. So we get a whole mix of different compounds in the body. And then there are the ones that were not absorbed initially because we could not digest them. They are transported through the body. They reach the whole distal end of our digestive system, and there they are broken down by bacteria. And here we see the way that they are broken down by bacteria is well studied. So the point I'd like to make at the end of my presentation now is that we know much about the structure-activity relationship of flavones and flavonoids, but only a small portion is actually absorbed into the body, and then they circulate for only a fairly brief time after we've eaten. And the amounts that are in the blood are not very high, but maybe we should look at these degradation products that are formed at the very distal end of our digestive system, and look at the action of these very small molecules that are derived from the flavones, and see if they have the same type of action, the antioxidant introduction, the activity on cell physiology. And so with that message, I'd like to thank you very much for listening to my presentation. Thank you.
Randy, thank you very much for your very nice presentation. I think we have one question. So I have a question for you.
Yes.
Yeah. Do you think the metabolites of flavonoids have some anti-cancer activity? For example, the quercetin? in your slide, you show many times about quercetin. If quercetin's metabolites can they be active compounds?
There are far fewer papers on the activity of these metabolic products. There are some, and I'm trying to get an overview of how much is tested on these molecules. I know that things like ferulic acid, which are also part of the diet, are small molecules, and they are tested and seem quite promising in some of the tests. At least as promising as the flavones themselves.
I see. Another is, for example, the quercetin glycide. Usually the glycide from the plant,
Yes.
for example, the lutein or quercetin or other glycides.
Yes.
But usually the glycide cannot enter into the liver and into the liver, the circuit system. Yeah, so most cases they were destroyed to glycogen.
Yes.
In the body. So how about if we directly, for example, inject lutein or quercetin glycide to the body? Can it be active compared with quercetin?
Yeah, it's a kind of engineering approach, isn't it? It is. You start with observation that some diets are good for you, other diets not so good, and then you identify some compounds, and of course you can find ways to increase the amounts of those compounds in the blood. But then maybe it works, maybe it doesn't. But you're not looking at the same mechanism, because you have to find an explanation. Why does a diet help? And obviously there are things in vegetables and products that may have pure medicinal value and can be developed as medicines, but it doesn't explain why they can be active in the low concentrations that we normally eat them. And that is really my main interest.
I see.
Right.
Thank you.
Thank you.
Thank you for a very nice presentation.
Thank you. And Professor Dr. Takashi Watanabe is the third speaker of this section. He's from the Kumamoto University.
Honorable Chair, I have a question. I raised my hands. So if you kindly allow me to ask a question. This is Dr. Atiya Rahman from Bangladesh. Would you allow me to ask a question?
Yes, okay. Yes.
Oh, okay. So thank you so much. It's really a very good piece of work from Professor Randolph. I congratulate him. By the way, I have a question that I worked on oxidative stress mitigation through using the medicinal foods. So before transfiguring medicinal or not medicinal, plant sources into a medicinal foods or functional foods. Yes. Could you please explain the inevitability to assay the biological antioxidative enzymes, right? For example, superoxide dismutase, catalase, and glutathione peroxidase. How much inevitable to check the upregulatory modes of this sort of enzymes before converting a plant sources into a medicinal food sources? Thank you so much. Am I clear?
Yeah, so you want to know at which concentration you need to be active before you consider them as medicines. Is that right?
Yeah.
Yeah, some of my colleagues, I'm in the School of Pharmacy, some of my colleagues work on medicinal compounds and they run test schemes, and typically they have this limit. If something works at 5 micromolar, then we consider it worthy of further investigation. If some of the activities or IC50 values are above 5 micromolar, it's not worth it, because really most compounds need to be active at sub-micromolar level, at nanomolar. So yeah, that's the limits, and that's something you often find in papers, which I detest a bit, is that people say good activity or excellent activity without giving a figure. So I prefer them if they give some measurement, some indication what they call good or what they call poor, because there are even papers that call 100 micromolar IC50 activity as good activity, and it's obvious that will never be will never have any medicinal value. So I hope that
yeah, thank you very much. I will connect you later.
Okay, thank you.
For more discussion. Yeah, thank you so
please.
much. Thanks indeed.
Yes, okay. I would like to introduce the third speaker, Professor Dr. Takashi Watanabe. He is from Kumamoto University, Department of Medicinal Botany. The title of his presentation is An Integrated GIS Study of Medicinal Barley from Turkey and Himalaya with Phytochemical Evaluation to Control Intestinal Flora of Human. I would like to give the microphone to Dr. Watanabe to make his presentation, please.
Okay, could you see my PowerPoint? Everybody can see it?
Yes, we can see. Yeah, no problem. It's very clear. Yes.
Okay. Thank you so much for the invitation, MESMAP. I really happy to open the MESMAP 6 because of around the world we have some issue of the COVID-19. Even though Dr. NAZIM is proceeded to this symposium, then I also joined this work. So now coming up to my presentation. So today's topics is an integrated GIS study of medicinal barley from Turkey, especially Turkish barley and the Himalaya, with a phytochemical evaluation of the control intestinal flora of humans. So today is a very shortly, I have to explain this. I have a 20 PowerPoints. So at the beginning, I just showed my work. So my major work is plant inventories. So field research on traditional medicine over the 35 years I spent the time, especially in Turkey. When I was in Kumamoto, so 5 years before, I just started joint work with the Turkish flora. Then I found many things from the Dr. NAZIM, and then so just to study in this time, I showed the Turkish barley specified. So you can see the first PowerPoint. This is my major work. Just have a look at the active ingredient chemical formula, and I used to go the field and making specimen. And after that, today I showed some calibration activities. So this is my major work. Therefore, I study about more in detail about the Turkish barley. So next page coming up. So I always talking about coffee belt is very important for the around the world, global, plant resources. Then north area is 25, in the south is 25 degree. So it makes, you have a look at the red color. We call the coffee belt. This is a major plant resources laid down around this area. Therefore, maybe this is a 66% out of all flora vascular plants. And we used to go to this area. But in this presentation, just touching the coffee belt around the Turkish area. But Turkish is very important, very special crops and the medicinal plants. It's a very famous area from the Europe, like Western medicines based on from the Turkish flora. So next one. Today I show the whole area for cultivations. For examples, this is the right-hand side, right, Kumamoto. Kumamoto is like middle of the Kyushu area of Japan. So that showed in red color, this is our cultivation area, 1 to 4. One is fast growing area, just leaf harvesting. This area is cultivation about Nepal Valley. Mr. Imasaka farm. This is fast growing area, cultivation area. And then second growing area is a Turkish one. This also Imasaka farm. The third cultivation area Nepal, from Nepal. When I was young, maybe I was 25 around, then joint work IPPGR. So I study about the valley of Nepal. And this seed we cultivate the third seed collection area, Mr. Noda farm. And finally, fourth cultivation area, the Turkey. This is Takayama farm. So the same time we cultivate study on in this year, the January, March around, I think. Then I show we compare with the standard with the Harushizuka. This is a Kumamoto local valley recommended Koshi city of Kumamoto. So today we compare with the whole different type of the plants. At the beginning, I explained something to the cultivation Hodeum classifications 1 to 11. 11 is now new type of machine. In this regarding this presentation, I wish to see the more new type of cultivars. So first 2 row valley. This is for the beer, using for the beer, main Europe. And the second, and the third is the valley, the 4, 1, 2, 3, 4 rows, and the holy Barigum subspecies Bulgaria, habitat in Turkey. And the 4 and the 5 is the 6 rows of valleys in the main crops. This name of the valley, Horma hexa stick corn. In the 7, Harus Valley, naked valley. This is naked valley. This is for food. When I was in Nepal, used to go to Tibet. So therefore, I collected this kind of naked valleys. 9, wild valley. This is the holy valley subspecies, spontaneous. So many Hodium, genus Hodium is laid down around the world, but especially Turkish valley is very, how do you say, rich zone of the valley because of this origin from the Turkey to the around the world. For example, this left-hand side, the Nepal valley, and right-hand side, like below the photo, have a look, Harushizuka, this Kumamoto. So this is the early stage of the March first week, and we cultivated, studied. Then we harvested only the green leaf for the using the our functional food. Then Nepal Valley little bit bigger, and the Harushizuka and the Kumamoto is more slims at the beginning. So therefore, this is good for the production for the only not using the seed, only for the green leaves. So next introduction is same way. Okay, so this is the 1, 2, 3, 4. This is the first Imasaka farm. It just showed using the with the IoT devices. We developed making ourselves from our laboratory devices. Their laboratories are very not expensive. We making the management ourselves for 5 years. And after that, we make the more concise, small size, and the more low cost make it. So introduction, Geographical Information System, GIS. So we used to use this GIS system because of this system is now very, making a more good new information from the plant resources. For example, this system can be modified for the researching for the individual, different type of small species too. We can use find it on the Earth, but especially in Kumamoto, we used to go the plant investigation of the medicinal plants. On the other hand, the GIS also can apply for the agriculture areas. For example, the biomass of the chlorophyll and another chemical component of the today is special for explain about the phenolic compound, flavonoids. And the GIS provide a study plant habitat condition by the integrated environment data. So many things, I just write it. So please have a look after that. So we used the multi-spectral camera specifications. This camera is very important for the plant, measurement of the plant diversity, not diversity, maybe biomass, and specify some chemical component from the valley. This brand is a blue, green, red edge near NIR, we call it NIR. This is the most important area for the plant growth. For example, have a look the wavelengths, right-hand side below, and then chlorophyll absorbs 0.4 to 0.7. This is chlorophyll absorption, but chlorophyll nearby the maybe 7 around, 0.7, 700 nanometers. Then, but the plant has a special behaviors, use cell structure we call the red color area, red edge. So this is involved the NIR zone. So this is most important for the plant growths. We are just getting information from this area too. So this shows the fast growing area, leaf harvest. Why we making the prepare the whole different type areas? First and second, only for the harvesting leaf, for the food, functional food, for the functional food. And the third area and the fourth area is we have to collect the seed. Therefore, we prepare the whole areas. This is the first. And this early stage of the March 3 in Japan. Now Japan is October. Today getting cold. But March also spring, early spring is quite cold. So therefore, we started the young stage of the harvesting this, green leaves needed. So this is Nepal, and the right-hand side is Harushizuka. You just have a look in the early stage is not so difference growing. And this is the second, fast growing area. Left-hand Nepal and the right-hand side is like Koshi cities, Harushizuka. And then left two photos is Harushizuka, and below there is Nepal one. So early stage is not so different for the growing, the young leaves. And then barley field data collection. Now this is third cultivation area. So this is only for the seed collections. So the early stage also not too much growing. So now we just put the scale 15 centimeters highest, not so big one. But after that, I just explained that this early stage is most important for the green leaf harvesting too. This is the whole cultivation area. This is only the seed collection from Turkey. And nowadays, we studied the sesame import from the Turkish. Turkish sesame also sending a lot to Japan. We import from the Turkish. So therefore, your Turkish food, this material is more important. I recommend that if we can investigate in Turkey, we are very, very happy to work together because of we found something new from the barley, but also we studied now the sesame too. So this stage is this area, the number 4 cultivation area, only for the collection seed. this is the first on the Harushizuka growing stage, the first week of the March, Harushizuka. So after we sowing, we started on the different stages for the every 1 month. Not 1 month, maybe every 3 weeks we go to the field. Then we catch up the multi-speak camera using the drone. We used to use it like Inspire Second. And this is a far field 1 Nepal UAV drone images. And we call the field 1 the RGB, blue, green, and red, and the NIR. We used to catch up this data. And also after that we calculations just below, have a look, body, visual atmospheric resistance index, TGI, triangle green index in the NDRE. So body is a whole investigation for the vegetation. TGI and NDRE is the chlorophyll contents. So you just have a C and different colors, but individual red and blue and blue, single color cannot see the color, only the black and white. So anyway, so this is a field 1 result, and this is another one. This is the second area, Turkish. This result using the drone. And the third is a Nepal one in the seed stock area. And this is a little bit difference, but I can show them in index. The Turkish, this is a field 4. So we are using the drone every 3 months to catch up the different, UV data. Then after growing the 2 months, after 2 months maybe, and we measured again. For example, I mean the January 28, January 28. So Turkish and Nepal, we just started sowing, and then March 2nd, 4, and then Turkish, and then after that Nepal also too, and then we compare with absorbance, 400, 300 nanometer, 450 nanometer, 660 nanometers. And the ratio of absorbance we calculate calculations. Then after 3 months, we finished the harvesting all of our field. Then January 2nd, January 200 this year, and the amount of early stage means green leaf. The amount of flavonoid is the first stage was high, and after 3 months, maybe 3 weeks, little by little go down to the flavonoid contents. In addition, in the sequence loss during the powdery process too. So we have to make take care about the powder process after the harvesting of the young leaf, because of we will big loss from the young leaf. After dry, we have to take care of the powder processes. However, the contents of loss varied between the varieties, and the Turkish variety having high retentions. So we compare with Japan and the Turkish. Turkish variety is more, benefit or big advantage rather than Japanese variety. But we have to study more in detail. So this is the barley field area data result. This is a field 1 to 4, and then different collection, data collection date, March and April, May and June. So last week June is mostly finished for all the harvesting of the valley in Japan. In Japan means in Kumamoto. So this is a high altitude, 20 meters. We used to UAV 20 meters, not more than 20 meters, because of more than 20 is a big wind. We come to, then UAV drone was effect, like not stable to flight. So therefore we used to measurement how much will be height and how much we flight. The average 15 minutes with this long flight. Speed like 3 and 2 to 3, and the pixel resolutions. Then the special sensor used is the central double OK lock and the ghost sensor with Centera. We call Centera. Centera has a multi-spectral zone and an NIR red, green, and blue. And then also we need a more specified real data. Therefore, we use the spectral reflection sensor, SIS and NI. We use the individual areas. So that we collect the NIR and the red zone only that one, 2 areas. Multi-spec camera can get the whole areas, but this specify this SRNI is only 2 areas too. So anyway, so this is the first primary.
Last 1 minute, Professor Watanabe.
Okay. This is a very primary investigation, but even though we are already know about the Turkish ballet is a very big advantages with the Japanese ballet, and also young stage is most important for the young harvesting for the plant flavonoid contents. So this is a procedure of the UAV camera imaging processing. So UAV up to the vegetation index. Vegetation index is using the not only the UAV, but we used to UAV and also feed IoT devices. And this is the future work in the remote sensing can see the chlorophyll content and the biomass. But most important, we are significant studies. We learn more about the chlorophyll contents compared with another functional chemical, component is a very, matching. So therefore, we wish to study more chlorophyll contents and the biomass. Thank you so much. Time is coming up. Thank you so much.
Thank you very much, Professor Watanabe. Is there any question to Professor? I think no question.
Quite difficult.
Okay. If no question, we invite the next speaker, Professor Yawarida Chulikikita from University of Konkan, Thailand. He will talk about the Miroestor attenuates the overlitema induced congenital impairment and brain tissue oxidation. Yawarida, you can start.
Okay.
Yes, we can see that.
Can you see?
Yeah, nice.
Full screen.
Okay, everyone, can you see my slide?
Yes, perfect.
Okay. Thank you very much, Professor Seo Jinbao, for the introduction, for introducing me. Good morning, everyone. First of all, I would like to thank Professor NAZIM, Professor Nek, and Sefki for providing this opportunity to present my research work. And the title of my research work is the effects of microestrol on the ovary ectomy induce the cognitive impairment and oxidative stress in mouse brain. This is outline for my presentation. Let's start from the introductions. In the present, as you know that we are going to the aging society, the number of aging population increase a lot in many countries around the world. And the menopause is a natural part of a woman aging process when the ovary begin to produce lower amount of estrogen, progesterone, and menstruation cysts. The menopause is classified into three states. Perimenopause, this one is typically lasts 4 to 5 years. The hormone begin to decrease and the symptom of perimenopause start. And the menopause, this one, the second state is the menopause. Menopause typically lasts 12 months, considered to be begin after 12 months of no period. And the last one, the last state is the postmenopausal. This one just remainder of the woman life. As you know that the symptom of menopausal present in many systems of our body. In the systemic, you will get the weight gain. In the skin, sometimes you can get the hot flush, dryness, itching. And for the psychological symptom, sometimes you can get dizziness, anxiety, poor memory, and inability to make the concentration, and depressive symptom, vaginal dryness, and back pain, soreness. So this time I would like to focus on the psychological symptom. So from this slide, you can see that the hot flush come with the psychological symptom about 70%. Okay, hot flush come with the cognitive dysfunction, insomnia. Okay, this one occur from the estrogen deprivation. After menopausal, the ovary cannot produce the estrogen hormone. That's why we get the estrogen deprivation condition. Estrogen play a very important role in the brain. Estrogen can normalize the neuron development, produce the neurogenesis, synaptoplasticity. Estrogen can protect the inflammation and oxidative stress in the neuronal cell, can preservation of the neurotransmitter activity, and reduce the formation and toxicity of beta amyloid. All of these important role of the estrogen in the brain. This one can help us to manage the cognitive function and memory formation. And the role of estrogen in cognitive function, if you take a look in the molecular level, we found that the estrogen can bind to the estrogen receptor subtype alpha and estrogen receptor subtype beta, ER alpha and ER beta. After this one form complex, this one can bind at the estrogen response element, and this one can stimulating the gene transcription, which lead to increase the cellular protein by increasing the neurotransmission, by produce the neurogenesis, produce the synaptoplasticity and synaptosis. If we talk about the gene that involved in the neurogenesis, BDNF, brain-derived neurotrophic factor, and cyclic AMP response element binding protein, we found that in menopausal women, they have the significantly decrease in the expression of BDNF gene and CREB. So from this one, this leading to the to produce the cognitive dysfunction and memory impairment in the menopausal woman. So from this one, we have many kinds of the symptom after you get the estrogen deprivation. How can we treat this symptom, such kind of symptom? The hormone replacement therapy is a FDA approved treatment to relieve the menopause symptom and can prevent the osteoporosis. It is used to supplant the natural hormone with the synthetic hormone during the menopause transition when hormonal production decreases. Okay, so from this one, the hormone replacement therapy can make the benefits, but sometimes there are several studies that suggest the long-term useful of hormone replacement therapy. This one can increase the risk of breast cancer, stroke, and circulatory side effects. So from this one, it is better for the researcher to find out the better estrogen. The phytoestrogen is the plant-derived that can mimic the estrogenic-like biological activity. In this slide, we show the phytoestrogen. Phytoestrogen is come from the plant. Maybe this one is safer than the synthetic hormone. The phytoestrogen that generally used is genistein. This one is from the soybean product. A randomized double-blind and placebo-controlled study found that genistein might have the positive effects on hot flush without a negative impact on endothelial thickness. And this one is the Puraria mellifica. The active component in this plant is myroestrol. Phase 2 study of Puraria mellifica demonstrate the great promise in the treatment of climatic symptom. And the last one is the resveratrol. From this time, I would like to focus in the phytoestrogen that from Puraria candorii variety mellifica. In Thai, we call Guao Krua Khao. Guao Krua Khao is belong to Leguminosae family. In traditional use, in Thailand, we use the Puraria mellifica or Guao Krua Khao for anti-aging, increase the hair growth, improve the memory loss, and increase energy, increase the blood circulation, and sometimes increase the appetite. This slide shows the chemical constituent that we found from the Puraria mellifica. Various chemical compounds such as chromium, isoflavone glycoside, coumestrant, and pterocaptan and sterol. Among of these chemical constituents that we found in Puraria mellifica, chromium derivative show the highest estrogenic activity. The member in chromium derivative such as myroestrol, deoxymyroestrol, and isomyroestrol. This is the structure of myroestrol. From the structure of myroestrol, you can see that the structure of myroestrol very close to 17 beta-estradiol. So myroestrol is a phytoestrogen that can mimic the biological activity of the estrogenic hormone. So from this one, we interested in the molecules of myroestrol. That's why the objective of this study, we would like to evaluate the effects of myroestrol on ovariectomy, induced cognitive impairment, and to determine whether myroestrol can produce the neuroprotective effects in OVX mice, and to compare the potential effects of myroestrol and 17 beta-estradiol in ovariectomy mice. Let's start from the phytochemical experiment. We start from the Puraria candorii variety mellifica. We keep bark root about 10 kilogram. After that, we isolated and did the structure elucidation, and we get pure compound of myroestrol. This slide shows the HPLC chromatogram of standard mixture between myroestrol and deoxymyroestrol. And this one is our plant extract. that contain small amount of myeloestrol and very small amount of deoxymyeloestrol. And this one is HPLC chromatogram of myeloestrol that we already purification. The content of myeloestrol and deoxymyeloestrol in the crude extract were quantified, and we get about 70 microgram per gram dry weight of the extract in terms of myeloestrol, and only 15 microgram per gram dry weight of deoxymyeloestrol. We use the OVX. OVX is ovary ectomy as the model of the estrogen deprivation. The mice were ovary ectomized by surgical to remove the both ovary. 3 days after surgery, we give them the drug treatment for 8 weeks. After that, we perform the behavioral test. After finish the behavioral test, the biological and neurochemical assessment were performed. And this table showed the group. We divided the mice into 5 groups. Sham operation, this one, no OVX, no ovary ectomize. And the second group is OVX, ovary ectomy, that give the treatment with the corn oil. And the third one is OVX, received 17 beta estradiol. And the next one is OVX, received myeloestrol in the dose of 0.1 milligram per kilogram IP, intraperitoneal route. And the last group is OVX, received myeloestrol in 1 milligram per kilogram IP. Okay, at first we test from the Y maze. This is the shape of Y maze. Okay, we put the mice at the one side of the Y maze test. Okay, and we let the mice exploring in the Y maze. Okay, the mice in general, the mice exploring in not repeated not repeated arm. So for example, if the mice exploring in 1, 2, and 3 in ordering like this, we give him 1 point, or 3, 2, 1, we give him 1 point. But if he exploring like 1, 2, and 1, he get 0 point. Okay, so from this one, we let them to explore in the Y maze about 8 minutes. After that, we calculate for the percent alternation. The percent alternation, in a higher percent alternation means the better learning and better cognitive function. The results show that in OVX mice, it showed the significant lower in percent alternation. It means that the mice have the cognitive impairment when compared with sham operation group. But after treatment with estrogen and myeloestrol, you can see the significantly increase in percent alternation, and myeloestrol show the dose-dependent manner. And the next one, we perform the novel object recognition test, or ORT. This one separated in 3 phases: habituation, sample phase trial, and test phase trial. So from this one, the objective of this test, we just would like to test that this mice can discriminate the familiar object and new object or novel object. If the mice can make the discriminate between familiar object and novel objects, so we found that he learned a lot and he can remember which one is familiar object and which one is novel object. So we calculate to be the percentage of discrimination index. And we found that OVX mice show the significantly decrease in percent discrimination index. But after treatment is estrogen and myeloestrol, this one can improve the cognitive and memory test. And the last one, we check the Morris Watermaze test. And the results show that, okay, after training, all mice show the tendency decrease in the time, escape latency time to find out the platform. Okay, and after 6-day training, okay, In day 6, we remove the platform, and the mice should, if he can remember the position of the platform, he can swim around the target quadrant. And OVX mice show that the swimming time in target quadrant is significant decrease when compared with sham operation group. But after treatment with estrogen and miroestrol in high dose, this one can improve the cognitive function. Okay, and we check for the uterus weight and serum E2. We found that in OVX mice, this one showed significantly decrease in uterus weight and serum E2 level when compared with sham operation group. But after treatment with estrogen can increase the uterus weight and serum E2. In case of miroestrol treatment, this one can increase only uterus weight but has no significant effects on serum E2. And when we check, we would like to clarify the mechanism of action that related to oxidative stress. Okay, we found that the OVX can induce the significantly in the oxidative stress. This time we measure the MDA content and nitric oxide level. We found that OVX can induce significantly increase in MDA and nitric oxide level when compared with sham operation group. But after treatment with estrogen and high dose of the miroestrol, this one can significantly decrease the oxidative stress in hippocampus. And the SOD activity and catalase activity also be performed in our study. Ovariectomy can induce the brain oxidative damage by decrease the superoxide dismutase enzymes and catalase enzymes activity when compared with sham operation group. But after treatment with miroestrol and estrogen, this one can attenuate the OVX-induced oxidative damage in hippocampus area. And we also check the mRNA expression, especially in the gene that related to neurogenesis, BDNF and CREB. We measure in hippocampus and frontal cortex. Okay, from this one we found that in OVX mice showed a significantly decrease in CREB and BDNF mRNA expression in both hippocampus and frontal cortex area. And after treatment with estrogen and miroestrol, it showed a significantly increase in CREB and BDNF mRNA in both brain area. So from this one, another mechanism of miroestrol, we show the molecular docking study. So from this one, we found that the miroestrol have the interaction in estrogen receptor alpha very close, very similar to endogenous hormone estrogen, as you can see from this picture. Okay, this picture is the miroestrol docking with ER alpha, and this one is isomiroestrol, and this one is deoxymiroestrol.
2 minutes, okay.
Okay, so from this one, this table shows the free binding energy and hydrogen bond. So you can see, you can observe that very miroestrol show the binding affinity very similar to 17 beta estradiol with very low free binding energy. Let's come to the conclusion. OVX can produce estrogen deprivation, and the estrogen deprivation can induce the cognitive impairment and produce the low serum level of E2 estrogen and can increase and can decrease the uterus weight and produce the brain oxidative damage. But after treatment with synthetic estrogen and miroestrol, this one can inhibit OVX-induced cognitive impairment and also OVX-induced brain oxidative damage. So from this one, we can conclude that miroestrol show the neuroprotective effects and improve the cognitive learning via this microestrol can bind at the estrogen receptor, very similar to 17 beta estradiol. Okay, and this one showed the improvement after treatment with microestrol, show the improvement of the cognitive dysfunction that induced by ovariectomy. And another mechanism is the inhibit the oxidative stress in OVX. Okay, this research, I would like to thank our research team in the Konkan University and also at the Japan University of Toyama and University of Shizuoka for their contribution work. And also thank our organizing committee for the great opportunity for me to share my work with everyone here. Thank you very much.
Thank you very much for your presentation. Are there any questions for?
Honorable Chair,
Okay.
This is Dr. Atiya Rahman. I would like to ask her a question.
Okay, please.
Yeah. So thank you so much. Really, I'm so happy to see that the working pattern and the protocol is completely resembling with the work we do in our laboratory. So in fact, I'm truly very much interested to make collaboration, because currently I have collaboration with Walalyak University, very active collaboration. Every year I visit there. So it would be very great if you are interested, I can write you to make collaboration. So,
yes, please.
Yeah, but just one thing. So did you measure quantitatively the superoxide dismutase, glutathione peroxidase and catalase in your hippocampus?
Yes.
You measured?
I measured the superoxide dismutase activity and catalase activities.
Oh, not glutathione peroxidase?
Not glutathione peroxidase, just only two enzymes. Yes.
Okay, surely I'm going to write you very soon. Thank you so much.
Okay, thank you.
You are welcome.
Thank you very much for the presentation.
Yes,
We will
thank you.
Next speaker.
The last speaker of this section is Associated Professor Dr. Elder Garaver from General and Toxicology Chemistry Department in Azerbaijan Medical University.
Hocam değişiklik var. Alban İbra ediyor.
Oh.
We are sorry. The last program. I would like to introduce Professor Alban from Albania, Vice Rector, new Vice Rector from the Albanian University. We are sorry for this change. This is the last change in the program. Welcome, and we are listening to you. Professor Alban, welcome.
Okay, thanks a lot, Professor Nazim. I'd like to thank you again for the invitation that you offered to me. And at the same time, I would like to thank you and your staff for this great organization in this very difficult time. Congratulations. Okay, I just presented. I don't know if you have seen my presentation. It's active, my presentation now?
Yes, very nice.
That's perfect. That's perfect. In my speech, I would like to present to you a part of the reality of medicinal aromatic plants diversity in my country.
Can you open your PPT?
I just opened. Did you see?
No.
I just share with you.
Yeah,
no. You should open it.
You need to open your PowerPoint, right?
Really? Really? Okay.
Yeah, open your PowerPoint and
Please
share.
close and open again.
That's perfect.
Share the screen. It's okay.
Yeah.
It's open. Yeah.
Yeah, it's okay now.
Not yet.
Not yet. Yeah.
It's okay now.
Yeah.
Okay, it's okay.
Perfect.
Now.
What happened?
Perfect. Yeah.
Okay, that's perfect. Okay, thanks a lot. I would like to present to you a part of the reality of MAPS diversity in my country, challenges for the future of it. Albania is a small country in the southeastern Europe, and as a small country, we have very rich flora, which is the reason that Albania has the very rich flora. We have a very favorable climatical conditions, which range from the coastal subtropical climatical conditions to the highland continental climatical conditions in the mountain areas and the alpine and subalpine at the same time. Geography, and you know that the Mediterranean region is one of the most rich region on the world for the diversity of flora. And at the same time, we are part of the Balkan Peninsula. Another reason is the many different types of landscapes that we have. And in this kind of landscapes are full of the diversity of flora. Albania has around 300 species, wild species. And this is about 30% of the Albanian flora. And we have 30 endemic species and more than 200 sub-endemic species. Around 10% of Albanian flora is part of the Red Book of Albanian. And normally, at the same time, Albania, it's one of the very rich country for the plant genetic resources. And in front of our estimated, we have estimated around 800 species that are considered as plant genetic resources for food and agriculture that came from the wild. And we are rich, and at the same time, with the species of MAPS, we have around 400 species belong of Albanian flora that occur for the wild. And the most of them are very important for our economy, which is the background of the MAPS in Albania. MAPS are very important, have been historically a very important role in Albanian economy. Not only for these days, but at least for more than 70 years. And MAPS are very important lines for the export in my country. And usually for any years, we export at least $30 to $35 million. And another very important point for Albanian MAPS are that current estimated are that over more than 100,000 families are involved in the sector of MAPS. And most of this family came from the area near of the mountain areas. And most of them are the pure family. Albanian has, it's rich, especially for the indigenous species of MAPS. And as we are full of the indigenous species of MAPS, this kind of MAPS are very, very important genetic resources for selecting the clones for creating the new variety of MAPS. A part of the background of MAPS in Albania is that Albania it's one of the world player in the supply of MAPS. And most of exports of Albanians are mainly to European country and USA country. And one of the very important species for Albanian is Dalmatic sage. And we call the Dalmatic sage as the queen of the Albanian MAPS, because Albanian is one of the very important exporter for the USA for the sage production. And in front of the estimated, more than 80% of all of the exporter that go to the USA have the origin from Albanian. In front of this situation, we see that, in front of this situation, we see that for year by year is increase the export of MAPS in my country. On this table, you can see that at least year by year, it's increased the quantity and the value in of the export of Albanian, and from 2008 to 2029 is increased a lot the quantity per year, and at the same time during the 2019 our export will go around $33 million. On this table, you can see the country of export of our maps, and you on the table is presented very clear that the very important partner for us are Germany, with around 3.5 thousand tons per year, USA with around 3,000 tons per year, Turkey with more than 1,000 tons per year, and France around 500 tons per year. And in front of these maps are very important for the economy. And as the interest of the foreign company is increased year by year in Albania, during the last 10 years, the cultivation of maps, it's one of the very important point of the strategy of our agriculture ministry for the future. Year by year, the cultivation of maps in my country is increased a lot, and actually we have around 6,000 hectares cultivated of maps in my country. And the cultivation is very important for the economy, but the most important is that they offer a part of the sustainable possibility for the incomes for the farmers. And another very big effort of the cultivation is that with the cultivation, we will do the best one for the biodiversity on the field, biodiversity on the nature, because we must decrease the pressure of the farmers to the natural area. And if we'll increase and if we'll offer the farmers the more facilities for the cultivation, they will stop the pressure for the harvesting for more than for many plants that are growing as natural in the natural area. And in front of the very important maps in the term of volume, I have calculated that the sage is one of the most important part, one of the most important plant for Albania. Another one is Juniperus communis, Hypericum perforatum, Satureia montana is very important, Uva ursi is very important, Rosa canina, Crategus monogina, and some other one. And now I would like to present to you the main product of maps in Albanian. And on this slide, you can see the most of them. And in front of this situation, as is increased a lot the production of maps in my country during the last 5 years, is increased a lot, and the level of the distillery in Albanian. Actually, in my country, we have around 12 distilleries with a capacity for more than 3,000 liters per year. And these distilleries distillate a lot of plants like sage, for example, like oregano. Juniperus oil is very important for the distillery in Albania, myrtillus oil, lauris oil, and helichrysum at the same time. And early production for the last year, 2019, it's around 36,000 liters. And normally this business is one of the very important for Albanian economy. Here you see the background of sage in Albania, and normally the sage, as I have informed you in the last slides, is very important for Albanian economy. And here you can see the more consistent, the chemical background of sage, and at the same time the essential oils and the compounds of them. And another very important plants for us is the Salvia triloba. And here you can see the essential oil background of them and the consistent of them. And another one is the Saturaea montana. And you can see the essential oils of Saturaea montana. And here on this background are presented the background of essential oils for Juniperus black one and the red one, Juniperus oxystratus and Juniperus communis. And another very important plants for Albanian economy is lavender, Lavandula angustifolia, and normally Lavandula angustifolia is very important not only like a herb for farmers, but at the same time the essential oils interest the interest for the essential oils of lavandula in Albania is increased a lot year by year. This is the reality for essential oils and the essential oils and for vaccinium and for myrtillus at the same time. Okay, I'd like to present it to you the next slides, a part of the reality of the problems that have the maps in my country. In front of this new reality that is presented in my country, in front of this reality of the increasing of the interest of many Albanian, foreign company for the maps production in Albanian. Our production has some problems. Our maps have some problems. And one of the big problems that we have in the maps in Albanian is the problem of the harvesting protocols. Early harvesting and the harvesting by removing the stock is one of the big problems that we have with the plants that are collecting by pickers in the wild, from the natural area. This is a big problem for us. And sometimes harvesters are encouraged to collect early, not exactly on the time, on the optimum time for harvesting. And if they will start in the collection in the early time, normally they will collect it without any kind of implementation of or respect of good agriculture and wild practices. And if we not respect the good agriculture and wild practices, normally we will go to the lost part of the genetic diversity, because if the second harvesting time, especially for sage, we're starting in the late summer or in the early autumn time, this will be the big damage and a big problem for the sustainability of the wild plants. And this is a big problem for Albania, especially for sage. And normally in Albanian, harvester or pickers in Albanian applied at least two harvesting time per year. The first harvesting time is in the late springer time, and the second one depend on the raining. It's in the late summertime or in the early autumn time. Exactly this second harvesting time is one of the big damage for the diversity and for the biodiversity of sage in a natural area. And one of the other big problem that we have are not only for sage, but are for some other plants like Sideritis rese, for example, or Oregano vulgarum, and at the same time for Gentiana lutea, that is one of the endangered species for all of the European country. Okay, normally, as I have informed you on the last slide, the harvester never respect the rules for the harvesting time of sage, and sometimes they end up cutting the whole plant, and normally this is a big problem for the sustainability of the plants in the natural area. And the other big problem that we have is that in these conditions, the habitat changing cross in the most part of Albania, and we are exactly in the very important moment, and we have very problems with eroded species and with the population level. And this is a big problem for us. And now we are thinking to find the solutions to stop this kind of eroded of the very important species. And normally this reality is not exactly the reality for this kind of 5 years but it's the reality of the last two decades in Albania. And actually, more than 68 maps are considered that endangered status, and 40 species of maps are included in the National Red Book of Albania. This is a big problem. Here's the list of all of the plants that are part of the Red Book of Albania. These are the list of the maps that are part of the Red Book of Albania. And you can see that the most of the important plants like Salvia officinalis or Sideritis cerezia or Organum vulgare are part of this list. And which is the situation now in my country with the ex situ conservation. In front of this reality in my country, in front of the genetic erosion that is present for more than 2 decades in Albania, in front of the pressure of the companies to have to increase the quantity year by year, we are in the very big problems. And in front of this situation, we have starting some programs for collecting of the seed materials for most important maps in Albania. And this kind of expedition started at least 10 years ago. And during this kind of expeditions, we have tried to cover all of the area in Albania. And normally we have collected the seed materials, and we have stored this seed material in our gene bank. For all of the people that have not a lot of information about the gene bank, I would like to inform you that Albanian Gene Bank is the national coordinator for all of the activity of plant genetic resources in my country. And these institutions, Albanian Gene Bank, it's part of my university, it's part of Agriculture University. And as the national coordinator of plant genetic resources in my country, these institutions coordinate all of the activity for collecting missions, for ex situ conservation and the in situ conservation in my country, which is the situation of the ex situ conservation of maps in my Albanian Gene Bank. Here you can see all of the geographical distribution of plant genetic resources in my country. You can see all of the signs are signs that we have been for the collecting missions in Albania. And during these missions, we have collected at least 489 accessions of the most important maps in Albania. You can see that, especially, for example, for Sideritis.
Doctor, last 2 minutes, please.
Yeah. We have collected a lot of species, a lot of accessions, and actually in our gene bank we have at least 150 accessions of Organum species and stage species. Okay, I will try to compare my presentation, and which is our strategy for maps. Our strategy for maps is primarily focused to monitoring, collecting, and study of genetic diversity of the maps. In our experimental field, we have started a program for the depth evaluation of genetic material of Albanian maps, germoplasm. And you can see on the right picture is the picture of our experimental field. And on this experimental field, we have started a breeding program to select the clones. And with the selecting clones, we're starting the breeding programs for creation of new variety with a high amount of essential oils and the favorable essential oils compositions to offer the Albanian companies and to offer the farmers in Albania the more opportunity with the new variety that came from our endogenous species. We have started this program for sage, and we have selected 50 population of sage collected during our expedition in Albania, and we have started a program for the depth evaluation of this genetic material. And at the same time, 4 years ago, we have started another programs with Organum vulgare. And we have collected 40 different accessions in Albania, 20 accessions of red organum and 20 accessions of white organum. And later we have planted there the seed material in our gene bank. We have used the randomizing block. And now we have selected around 65 lines of oreganum. And very soon we will have the new variety of oreganum creating from the researcher of my working groups. Which are the problems which are the results and conclusions now for the reality of the genetic material of medicinal aromatic plants in Albania. MAPS in Albanian play a very important roles, and normally the cultivation of them are very important, very effective means to promote and to offer the farmers the more possibility to have the sustainable incomes, and at the same time to stop the pressure of the pickers on the natural area to protect the biodiversity. And for this reason, we have starting the programs for the deep evaluation of the genetic materials, and with applying of the genetic improvement, we'll select the clones and we'll create the new variety of MAPS. Okay, these are a part of our diversity of maps, and now thank you for your understanding. I try to do the best one to respect the time.
Thank you very much, Dr. Alban Ibraliu. It was very nice presentation. And is there any question to Doctor? So I think no question. Unfortunately, we are at the end of the first section of MESNAP 2020. I would like to thank to all speakers and audience for their participations. For second section, I would like to turn the microphone to Dr. SEKEROGLU. Microphone first, SEKEROGLU.
Thank you, Professor Aytemir. I would like to thank you all the invited speakers, Professor Yusuf Baran, Randolph from UK, and Watanabe from Japan, Yaoret from Thailand, and Alban, my dear Alban, from the Albania, and medicinal plants, the main importer from Albania, Turkey, and we process and re-export and use in Turkey. We are the same climate, and we are the same culture, near close culture.
Absolutely.
We were in Adriatic, near Adriatic with Alban. I hope so. After COVID, you will visit them. Also, you want to visit UK, Japan, Thailand, and other countries. So we are sorry we cannot service you coffee.
Professor NAZIM.
Coffee and fruit. Please.
Can I interrupt you just few seconds? You sent me a message to chat box. I replied over there. The important issue is that you invited me, but I'm very much happy. But due to some inconvenience in Izmir, I already booked a ticket this 2:38. I will start from here by train to go to Istanbul. So if I'll be very much grateful if you make my schedule before 2:15. Anytime is fine for me.
Oh, okay. We can request from Professor Aytemir. You can make your presentation. Mutlu Hocam duyuyor musunuz? Mutlu Hocam. I should ask Professor Mutlu if it's possible. You can make the second presenter in this session.
Okay.
1 minute. So we will chair this session with Professor Takashi from Japan. Takashi, Professor Takashi, are you on there?
Yes, okay.
Please, you can start the session and invite Professor Jumbo from China. You can start the session.
Yeah. I wish to see some schedule. I'm looking for that one schedule.
Professor Atiyah Rahman, I will contact you.
Okay, thank you.
I will write you. Okay.
Jumbo-san. Okay.
Hi.
Hi, Jambu-san. We are now Watanabe speaking. So please, your presentation fantastically. So maybe we are looking for your good presentations.
Yes.
So you introduce yourself, okay? So now you're, yes.
Yeah. Can you see?
Yes, it's all right. Everybody see that one. So let's start. And then we have you have a 20 minutes, right?
Yes, perfect.
Okay. Just start it, please.
Good morning, everyone, ladies and gentlemen. It's my pleasure to share my recent work with you. I think the organizer gave us a chance to gather on the internet again. So today I will talk about the stability of quercetin in the DMEM medium. I'm Jianbo Xiao from University of Macau, China. So now in Macau, in China, we are very safety. We can travel everywhere. So now I'm in mainland of China, not in Macau, but we can freely travel now. So we hope Europe and other countries can also can overcome this situation, can travel. So next time we can meet in face to face. Okay. So polyphenol is very important, and many people talk about it. For example, Randy from UK, we have many every year. We have many papers, many research on this area, and have many activity. I don't want to talk too much. And I think in most cases, the most important one thing is not polyphenol, not too much polyphenol become drugs. I think the most important is prevention some disease, I think. And also the cell expansion is very popular for the polyphenol bioactivity study. For example, anti-cancer like that one, we incubate the polyphenol with some cells for 24 to 72 hours, then see what happened for the cells. However, not too much people talk about what happened for the molecules you added. Means polyphenol or small molecules you added in the cell culture. So we first studied the polyphenol in the DMEM, studied the stability. So here we just simply report that after we add the polyphenol to the DMEM without any cells, we can calculate the structure activities. This slide we showed that this for compound myostatin, quercetin, camphor, and glangan, different in the ring B have different hydroxyl group. We can find that myostatin is very unstable. Within 10 minutes, it's almost disappeared. But quercetin, within 2 hours, we still can detect something. So here we just report quercetin's some result. So why the quercetin disappears so quickly? So then we investigate what happened in the PBS. PBS is I think most important buffer when we do some biological study. So we add the quercetin to the PBS only 5 seconds. Then at minus 20 degree methanol. Then to centrifuge, then use the UPLC mass method to detect what's happened. So here we find that within 5 seconds, the quercetin, half of quercetin was changed to its, I think, a new compound here with the mass to charge is 600, almost twice of the quercetin. In methanol, it's very stable, but in the PBS, suddenly reduced. So then we use an institute UPLC mass method, means we add the quercetin to the UPLC just add then directly at the DMEM, then set up all the UPLC mass method. Then we use the auto sample as a reaction. So means use auto sample to control the temperature, then to detect what happened with the increase in time. So maybe every 50 minutes we inject one sample. So in this case, we will not use centrifuge, not to precipitate, and I think can directly see what happened during this incubation. So here we can find if we use this method, we can find this is quercetin. Quercetin is almost disappeared about 10 hours incubation disappeared. In the same time, some new compounds formed during the incubation. Some was disappeared again, and some was formed. So it means during this incubation, many things happen for the quercetins. Let's see. So then we investigate the possible structure of quercetin in the DMAM. Here we analyze its dimer of quercetin, and then it formed at the water, then oxide, then it was degraded, and many things happen. We identify the possible structure. And also we can find that this slide shows the quercetin without the ascorbic acid. We see we found the quercetin is a little bit after 240 minutes it disappeared. However, if we add the ascorbic acid to the DMEM, the quercetin can be much more stable. So it means the ascorbic acid can improve the stability of quercetin. Here we can find that this is we calculate what's happened for the quercetin in the DMEM. First, it's add water, and then to its dimer, then oxide, then degraded, and many things happen. So during this incubation, so just now we talk about things without any cells. Now let's see what's happened if there are cells. We want to see what's happened if we have many cells. So here we test several cells use quercetin, incubated for 24 hours to 72 hours. So here we can find when we incubate with different kind of cells, the metabolites are quite different. For example, in A549 cells, there are many metabolites, but for SK28 cells, there are only two kinds of metabolites. Let's see what's detail. So when quercetin incubated with A549 for 48 hours, we can find that there are many metabolites formed. Most are its glucuronide and sulfate. And the main compound is its oxidized quinose metabolites, not the original one. We can find the mass to charge of 478, and it is oxidized quinose metabolism, not its original one for the A549 cells. However, when quercetin incubated with SK28 cells for 48 hours, we can find that the metabolite is methylated compound, means at a methyl group, and also it was oxidized. But we don't know it's first be metabolism, then be oxidized, or first be oxidized, then be metabolized. We will discuss it later. When the quercetin incubated with MCF7 cells, we can find that the metabolite also it's oxidized methylated compound, not the original one. But we still find some other metabolites of quercetin in the MCF7 cells. For the CAR2 cells, we also find that there are so many new metabolites when incubated the quercetin. Most are the oxidized quinol of quercetin. So for example, the glucuronide and the sulfonide and the methylated form. So here we summarize that quercetin in cells, it usually can produce some metabolites. Most of the cases they are oxidized quinose glycosides, for example, sulfate, like the unmethylated compounds. So how this form? Because this is 48 hours incubation. So now we want to use more cells to investigate how these metabolites form during the incubation. So we test what's happening inside the cell. We just remove the medium, then collect the cells from the flask. Let's see. Here we can find that within 30 minutes, we can find inside the cells, there are so many metabolites formed when quercetin added to the A549 cells. So because the quercetin have I think we have the 4 position can be glycide. It's 7 position and 3 prime, 4 prime and 3 position. So means here have the 4 isomers during the incubation. So means quercetin can very quickly enter into the cell, very quickly be oxidized, be metabolized in the A549 cells. Also, with the increase in incubation time, we can find that some new compounds formed, but the mass is less than its metabolism. So we think that during the incubation, so the quercetin glycide was oxidized to its quinose glycide. So with the increased incubation time, we only can detect its oxidized quinose metabolites, not the original compound. However, in MDA-MB 231 cells, we can find that with the increasing incubation time, yes, within 30 minutes, most quercetin can enter into the cells, and then inside the cell it was oxidized. But we cannot find some other metabolites. For example, it's glycide, sulfate, and like this one, only oxidized. So means in MDA-MB 231 cells, it was oxidized inside the cells. Then we check what's happening in the HepG2 cells. HepG2 cells, we have many kinds of metabolites. So we can find within 1 minute, some quercetin enter into the HepG2 cells. Then from the 2 hours, some metabolism formed. I think first it's methylated compound, a methyl group, and then oxidized, and also we found it's oxidized quinol compound. Yeah. So let's see some medium of the HepG2 cells when we added the quercetin. In the medium, we also find the similar result that in the medium, within 1 minute, so the quercetin was oxidized, and then after 4 hours, some metabolites was excreted to the medium. For example, it's a methylated compound of quercetin was drained out to the cell. This oxidized, this is methylated and oxidized compound. We can detect in the medium. Yeah, it's from the cell. And also, we can find that with the increased incubation time, some more quercetin metabolism informed inside the HepG2 cells. So here we can tell when we add the quercetin to the cells, so many things happen. First, it's oxidized in the medium, then enter into the cell. Also inside the cell be methylated, oxidized, and also be metabolized like and methylated. So many things happen. So when we add quercetin to the cells, and also we can find that ascorbic acid can improve the stability when we improve with incubated with A549 cell. Here we can find without cells, we can find that within 8 hours, so most quercetin were oxidized. But when we add the ascorbic acid to the cell, we can find that in 10 minutes, still we can detect its quercetin dimer. But without ascorbic acid, we cannot detect its dimer. And also within the ascorbic acid, the quercetin is very stable. After 80 hours, it's still high. And also we find that the ascorbic acid itself didn't inhibit or inhibit the F549 cells, but it can increase the inhibition of quercetin against these cells. So means ascorbic acid improved the stability of quercetin, which enhanced the inhibition activity. So conclusion is that quercetin unstable in the DMEM medium, and to form the dimers in PBS. The dimers easy to oxidize and finally degraded in DMEM. And the metabolites of quercetin are different in different cells, are quite different. And ascorbic acid improves the stability of quercetins. Thank you for your attention. So I will very quickly to share my work. If you have any questions, can discuss.
Some, Professor, have a question or not? Please, hang up. Hand up, please. No?
If I can ask a question, it's Randolph Farouk. It's wonderful to see all these metabolites, but how small a sample can you still analyze? Because you work with cell cultures and then have to analyze it and deduce the structure.
Yeah. No. I know for the cell culture, I just did directly to detect. I didn't extract. So I just use the method directly detect, because now we seeded the cell to the, how to say, so we seeded the cell in the we seeded the cell in this tube then use this UPLC auto sample as how to say, an incubator, so it can directly detect what happened inside the culture. But for the cell, we also remove the medium, then take out the cell, then to do medicines to extract. Yeah.
Thank you.
Okay, Jambos, I have some questions. So I was working on the plant tissue cultures, converting the glycolizations. So why are you using the cell cultures instead of the plant tissue? Like this is a safety making some of your drugs or not?
Yeah, because many people use the cell model to do, for example, anti-cancer experiment or many experiment. But we find that in the medium, in the culture, many things happen. Exactly, the cell experiment is not enough to investigate some result, because you don't know what's the bioactive compound. So I do this experiment just to tell that you should not do cell experiment. It's not the price. You don't know what's the active bioactive compound. Yeah, when you add some small molecules, it's immediately changed. You don't know what's the really bioactive compound. So we want to like this one. Yeah.
Okay, thank you so much. So maybe time is up now, coming up. Yeah. Okay, thank you so much, Professor Jambu.
Thank you.
Thank you so much. And then now, second invite lecture. Can I proceed to myself? Okay, introduce.
Yeah.
And Professor Dr. Mutlu.
Takashi.
Yes. Yes.
Now we changed the Professor Atiyar Rahman.
And now,
Professor
okay.
I can introduce him, Professor Atiyar Rahman from Bangladesh. Now he is in Turkey. He is in Izmir. Only one participant face-to-face in Izmir. We are sorry for this inconvenience, but our government have some precaution. We couldn't manage hybrid congress in Izmir. Now he is in Izmir, and but he is originally Bangladesh. And he is our the second invited speaker this session. Please, you can start, Professor
Okay.
Atiyar Rahman.
Please go ahead.
Please open your microphone. Atiyar Rahman.
So could you allow me to share my slides, please?
We couldn't, we cannot, now we can see you.
I got it.
Please share.
Okay. Just a moment. Let me open my.
Okay, very. If you want, you can send and we can share from here, Professor.
Yeah, actually, let me. I downloaded the file, but it's a bit difficult. PPT.
If you are not available, we can start another speaker and then you can make presentation. Professor Atiyar Rahman.
Okay. So
okay.
So I'm preparing my. So next one I'm going to.
Okay, you find, and I am inviting now Hari, Professor Hari Deukota, Hari-san from Japan. Can you hear me?
Yes. Hello everyone. Yeah, I'll just share the slide. It was just saying so.
Yeah.
Just a minute.
Okay. Now,
Atiyar Rahman, please close your sharing. Please cancel your sharing. Hari-san, it's okay. Welcome, Hari-san.
Yeah, thank you. So I hope my slides are visible now.
Okay, now okay.
So good afternoon, everyone. It's already evening in Japan. So I'm Hari Devkota from Kumamoto University, but I am from Nepal originally. So for the last 10 and few more years, I'm living here in Kumamoto now. So today I would like to present about plant phenolics, antioxidant and enzyme inhibitors, and their characterization and bioactivity analysis. So as the previous speakers have told that medicinal plants are very, very important for our health. And the WHO report also says that almost 80% or more than 80% of the world population, they depend upon medicinal plant as a primary source of healthcare. And these medicinal plants are not only used as these plants are not only used as a medicinal plants, but we also use them as a food, as a cosmetic and spice, food additives, functional foods, and there are many. And this is a report that was published in 2017 by Royal Botanical Garden in Kew, when they said that only about 28,000 medicinal plants out of 452,000, there are only around 6% of the world plants they are used as medicinal plants. So there are many plants which we need to explore. We have to study more about the chemical constituents, and possibly we have to study them more. Probably they can be a lead for drug discovery, or maybe we can develop functional foods or cosmetics. We have to study more on that. And these are the few drugs that are developed from the medicinal plants. I don't want to go much in detail about this, but only one important thing is that even we say that natural products are very, very important in drug discovery and design, and especially they are important, especially for the anti-infective agents and anti-cancer agents, because they say the report says that more than half of anti-infective and anti-cancer agents, they come from the medicinal plant. But if we see the overall drug discovery, we can see that only this one, which is in the green and this light green, only about 33% of the total drugs they were reported in last almost 4 decades from 1980s to last year. They were of the natural product origin, and that includes not only plants, but the microorganisms, fungi, and other things. So, but now is the new coronavirus infection is also there. We also need, of course, we need vaccines. We need other antiviral drugs also. But there are many studies there now focusing on the natural products, or especially plant phytochemicals, as a treatment option for the COVID-19. But today I will basically talk about the medicinal plants and functional foods that work we're now doing in Kumamoto University. A part of this is also in collaboration with the current session's moderator, Professor Watanabe. So in recent years, there is a significant increase in the development of plant-based functional foods, and they are for the maintaining proper health condition and prevention from the diseases. But in many cases, when these functional foods or nutraceutical products are advertised, they are said to be very good for the health. But in many cases, we do not have sufficient in vivo experiments to support that, or there are very few clinical studies who suggest that. And in many cases, the actual chemical constituents of these plants or functional foods, they are the main active agents. They are not reported, and even they are reported, maybe they show the very strong activity in vitro, but they are very weak or not active in clinical studies. Also, there was what Professor Ronald Aru presented in the second presentation today. So, but in this, I will speak very basic about our research in Kumamoto to identify the chemical constants and in vitro activity of two plants. What is this? One is this Lindella sericea, family Lauraceae, and another one is this Farigiphium japonicum, and it has a variety one called Gigantium. I will talk about these two plants. So first one is this Lindella plants. Today I will talk about this Lindella sericea. In Japan, also the term functional food also started from Japan around 1980s, and now in Japan there are many functional food that are in market, and one of them is like this is in Japan we call it kekoromoji. So this is the Lindella sericea. The leaves and also the stems and wood part are there used as a functional food or health, especially as a tea. And traditionally in China and Japan, they also keep the stem part in some of the liquors, and they say that it's good for health. But there has not been any study on this Lindella sericea, and there have been few studies on other species like Lindella umbellata or even its variety, the glaberata. But there was no study in Lindella sericea. But few products are marketed, so we just wanted to see the chemical profile, what are the chemical constituents present there. So in our study, we first we are screening many medicinal plants that were in our garden in Kumamoto University and also in Kyushu area here in Japan for their in vitro activity assays. And one of the assay was alpha glucosidase. Sorry, this is glucosidase. It was my mistake. Alpha glucosidase inhibitory activity, and also did antioxidant activity by DPPS method. They are very basic assays, but when you are screening hundreds of extracts, maybe they're easy to do. And we found that Lindella sericea extracts showed very strong alpha glucosidase inhibitory activity. But one limitation in this case was that the alpha glucosidase we had was from the Saccharomyces cerevisiae, so it was not from the animal source. Maybe the activity of acarbose is very weak in reported, but still we had some activity for the extract. And in antioxidant activity, this positive control was Trolox, so extract was not that much active, but still we continued the chemical isolation. And this is the flowchart what we did in chemical isolation. We used dried leaves and twigs about 2.5 kilo, and we extracted with methanol, and the extract was then subjected to repeated column chromatography and MCI gel. This is a polystyrene gel, and then these fractions were further subjected to either Cepadex, ODS, or silica and other columns, and we finally obtained 8 total compounds. The structures of these compounds were identified using NMR, basically NMR and mass. So one of the compound was this one. This is a flavanone. This compound pinosembrin. So this is a proton NMR and carbon NMR. I'll not go much in detail about the structural elucidation today. And another compound we identified is I have assigned number 4. This was also a derivative. Here we can see that around in proton NMR around 3.8. There is one peak that is especially for the phenolic methoxy group. So on this basis, we And also on the basis of NOE spectra and 2D spectrum, we decided that we confirmed that this methoxy group is attached in the 7th hydroxyl group of this pinosembrin. So this compound is pinoestrobin. So similar, we can also see the comparing the pinosembrin NMR data and pinoestrobin data. We can see that there is one additional signal in carbon NMR for the methoxy group in 56.3. So like this, we confirm their structures. So all compounds that were isolated from the extract of Lindella sericea were epicatechin, minus epicatechin, taxoprolene 3-O-glucoside, pinosembrin, pinoestrobin, and pinoestrobin chalcone. This is the chalcone form of this compound, and quercetin. It's a quercetin amnoside, and quercetin 3-O neohesperidoside, and quercetin. These were the basically all were the flavonoids we isolated. Then we just wanted to check that what may be the active compound that showed good antioxidant activity in vitro. So this was the extract data. And then we found that this is because we had the extract. This is in microgram per milliliter IC50, and this is in millimole. And here almost all compounds except 3 and 4, they showed good activity. And this is very common because if I go back to the structure, this compound 3 and 4, they do not have any hydroxyl group, especially in the bearing of this flavonoid. So this type of compounds are said to have not good antioxidant activity. So these are the other compounds which, and most of them had the catechol group or catechol moiety in the bearing. So obviously their activity is relatively strong. And also we using this same Saccharomyces cerevisiae derived alpha glucosidase. We performed the alpha glucosidase inhibitory activity. And if I see the IC50 value here, the highest activity was shown by the quercetin, and the quercetin was weak compared to quercetin. Maybe it's because of the glycosylation here, and some other compound, especially compound number 2, taxoprolene 3-glucoside, showed some activity. And here also acarbose looks very weak. But we also did, I don't have final data here, but we also tested on the rat intestinal powder that has alpha glucosidase, the mammalian source. We also did that experiment. But in that case, acarbose was very, very strong, and our compounds are very weak, more than 100 times weak. So we are now preparing the compilation of this data, and then also we are trying to show that even in vitro assays, in especially the enzymes that are obtained from mushroom or other, especially non-primates or other organisms, sometimes maybe their activity in vitro looks very strong, but they may not have the same activity in other source enzymes. So this was about the one plant. The second one is this plant Fragarium japonicum giganteum. Many plants of the Asteraceae family, which was previously also known as a compositae, these plants are widely used all over the world as a food or medicine, especially like marigold, and there are many flowers that are also used as a food and a health supplement. In Japan, this is not a variety. This photo is of the main one. This is Fragarium japonicum. This species has only two. This genus Farbigium has only two species. One is japonicum, another is hibernifolium, and hiberniflorum. Sorry, and among these, this Farbigium japonicum has 4 varieties, and this is a Japonicum giganteum and Luchense. They are mostly found in Japan, but this Formosanum, this is mostly distributed in Taiwan. So there have been some studies on other varieties, but there was no study on this variety giganteum. And also there are no studies report for the flowers. Especially there are few studies which report the essential oils and their activity, but there are no studies on this species and its variety for the other non-volatile compounds. So I obtained this plant from Professor Watanabe, and we did the chemical analysis, like in case of earlier plant. And this is, we used dried 1.5 kilo of the flowers. We extracted with 70% methanol, and we isolate total 5 compounds. And this is the NMR data. So this is the rosmarinic acid, very common compound in the flowers, especially of Asteraceae family, and also in many other plants, especially the Levity family also. And but comparing the structure of the rosmarinic acid, the another compound we obtained is petacyphenol. It is a decarboxylated derivative, and here you can see the one ketone group is there. So this compound is not new, and it was reported in around 1990s from another plant of the same family. That's also very commonly used as a food in Japan. But this is the second plant source for this compound, and only second time reported from the plant source. And this is the carbon NMR data. And these are the total compounds we isolated: the beta-cyphenol and the rosmarinic acid that I already explained in the NMR. And then we had 4,5-diocapalgonic acid, 5-capalgonic acid, and quercetin. And of course, these compounds are very, very common, and even in the chrysanthemum morifolium or other flowers that are edible from this family, these compounds are very common. But in our study, it was the first study, and the flowers themselves are not used as a food source for this plant in Japan. And that's why we try to evaluate some in vitro enzyme inhibitory activities. And this is the antioxidant activity. I will not go much in detail about that because I already explained for other plant. And we also evaluate the tyrosinase inhibitory activity using the mushroom tyrosinase as a model. Sorry, there is some problem with the font. So arbutin was used as a positive control, and we had mushroom tyrosinase. And in this, and L-tyrosine was used as a substrate. And here we found that, especially the compound number 1, the beta-cyphenol, it has a very strong tyrosinase inhibitory activity. And of course, the model organism or model enzyme may be different, but still it was quite strong compared to the data of arbutin and also of quercetin. So we hope that further study is necessary to confirm the activity of beta-cyphenol, and also, of course, we need more study on the toxicity and other things. But maybe we can further search if beta-cyphenol is a strong tyrosinase inhibitor that maybe can be used if it has a sufficient, we can obtain sufficient data on its effectiveness and efficacy, and also on the safety. Maybe we can develop this one, or maybe the plant extract as a whole. Here extract was not that active, but maybe this compound we can develop as a material for cosmetics, but that warrants the future research. So this is the conclusion. So we isolated a few flavonoids and phenolic acid derivatives from these plants, and in vitro system they showed some strong activity, and especially the potent tyrosinase inhibitory activity of beta-cyphenol is interesting. So maybe we have to study more in future as a cosmetic ingredient, and detailed animal model and clinical studies, they are necessary, and to show their effectiveness in vivo systems. And also we need studies related to their pharmacokinetics and safety. For example, even I was talking about the beta-cyphenol. So even we want to use as a cosmetics, or we hope that it will have some activity and to treat hyperpigmentation. Maybe we have to look on its data. Can it really pass through our skin or not? So this type of studies are necessary in the future. So this is all about my presentation. Finally, I would like to show some photos of our medicinal plant garden in Kumamoto University. So this is very difficult time. So we are participating from our countries, but in future, we also are hoping to welcome you in Kumamoto University. This is our university school of pharmacy here, and this is our medicinal plant garden. And we are working on medicinal plants of many countries. So this is our museum of traditional medicine. So I really hope that in future and sometime we can welcome you in Kumamoto and have further discussions. Thank you.
Thank you very much, dear Harisan. Is there any question? No question.
No question.
Takashi-san, I will introduce Professor Atir Rahman.
Okay. No. It's okay.
Yes, I will share.
Okay, program.
Atir Rahman, please close your. Can you share, Atir Rahman? I can share here.
Okay. Thank you so much.
Sure.
Yeah.
PowerPoint maybe.
Kapat Atir Rahman'ın şeyini.
Yeah.
Kapattın mı? Ben ne görüyorum? One minute.
Okay, come on.
Now it's okay, I think.
Okay. Today's special topic is the Alklaman, standardized Licia glutinosa upgrades expressions of the oxidation, right? And the enzyme and the normalizing levels to the control diarrhea incidences. Okay, so please go ahead, Amansa.
Yeah, thank you so much, Honorable Organizers. I'm really delighted to be here to present on the topic as you told. Hope you can see my presentation. Slides, right? Hello, can you? Yes.
Yeah.
Okay, thank you. Yeah, thank you so much.
Don't worry.
Yeah, actually, I must congratulate and I must show my utmost gratitude to the organizers. I'm only the presenter who is in Turkey from Bangladesh, only because of some miscommunication. By the way, let me present. So our presentation actually goes on first to introduce the standardization procedure of plant material. Before that, I must clarify that why we have chosen this sort of topic on antidiarrheal. Because we know that according to World Health Organization, millions of people over the globe have been suffering from diarrhea from different causes. I mean, the etiologies of diarrhea are different. And that sort of diarrhea, actually, we so far consider usually the bacterial diarrhea, excessive consumption of alcohol, sometimes intestinal disorder, oxidative stress, then overeating, then some other causes, for example, water consumption not sufficient. So this sort of diarrhea makes us compelled to use different sort of antidiarrheal drugs. For example, loperamide, this is a standard drug. So far we use. And among all other antidiarrheal drugs, we are very much experienced of different sorts of side effects or adverse effects we can see. And among them, the loss of appetite, stomachache, in the nausea, vomiting, drowsiness, sometimes skin rashes. These are very common. So since long been in our country, particularly in South Asia, China, India, we have been so much accustomed to use traditional medicines. And based on that concept, we use this plant, which is long been used as antidiarrheal plant. However, no scientific evidences are available on that plant, which is called Lichia glutinosa. There are some Bengali name or local name in our subcontinent. Hope you will see in my presentation. And particularly the bark of this plant, roots, and sometimes the leaves are very much available to use for diarrhea. So our objective is to find out the antidiarrheal ingredients and the mode of action, how they work. Could you please go for next slide? Next slide, please. Yeah. Next one, please. Yes. So, as we said that the main focus, how the mechanism is lied on over this sort of antidiarrheal effect of the plant we call the Lichia glutinosa. So before going through that, many of the people, they use the plant material, but they don't standardize. So we standardize controlling different sorts of parameters. I'm not going to detail, but temperature, then the environment, the humidity, and the timing. Using this sort of parameters, we standardize the plant materials. And that standardized plant materials is compared with the non-standardized plant material. In context of the production, I mean the yield of the plant material, their activity, and the plant phytocompounds we collect and extract from the plant materials. So Could you, yeah, next one. So this is in short, no, yeah, this is fine. So this is in short our whole protocol of the work. We collect the plant material and use the conventional procedure to isolate the, extract the crude extract, and that crude extract is subjected to go for phytochemical analysis. Before phytochemical analysis, we do some qualitative analysis. Among the qualitative analysis, we use the total phenolic compound, then total polyphenolic compound, flavonoidal compound, make a GCMS spectra, then the presence of the phytochemical secondary metabolites, alkaloid, steroids, flavonoids, and all others. Confirming that, we go for the TLC, SPLC to collect the, isolate the compounds, and those compounds are identified through some subspectroscopic analysis. Once they confirm the crude extract is allowed to go for in vivo work, then we start the in vivo. Before in vivo work, we must confirm whether the sample is toxic or not through acute toxicity test. Once we confirm that the plant material is not toxic, then we apply this for an in vivo model. After in vivo model, we justify and evaluate the same data using the bioinformatics tool and some in silico model. So in combination with all these, in association with the molecular study, for example, the mRNA expression of the particular proteins or particular genes, we finally confirm how this mechanism is controlling over the antidiarrheal potential of the plant material. Next, please. Yeah. So this is, I'm not going to detail it. This is our conventional procedure, how we extract the plant material. The crude extract. So we can go for next page. Yes. This is very important. Although the issues are very simple, but important, because nowadays scientists are focusing more on the oxidative stress. They are saying this is the pivotal factor which can lead to cause a lot of diseases, not only diarrhea, diabetes, cardiovascular diseases, then neurodegenerative, and a lot of diseases. So this is truly inevitable to check whether the plant material does have very significant antioxidative activity. So we have a lot of antioxidative in vitro protocol. I'm showing here only 3 or 4. In these protocols, we check the superoxide scavenging activity and iron chelating activity, as well as their phenolic content and the flavonoid content. And we see from the data that superoxide scavenging activity, iron chelating activity, there are some certain values we call the cutoff values. If the cutoff values is less than 1000 microgram per ml, in that case, according to the phytochemists, this sort of phytochemical, the plant extract is viable to go for the further experiment for animal models. So in our cases, we found that the superoxide scavenging activity of the extract is 141.12. It does mean it's truly valuable and significant and usable for further processing to be used in vivo models. So similarly, in iron chelating activity, we found the extract, it does have the IC50 value 128.013. So based on this in vitro antioxidative data, we went to go for the next acute toxicity test for a prior criteria to use it in vivo model or animal model. So could you go for next slide, please? Yes. So this, according to this protocol, the animals randomly selected for a week, and the doses are increased up to having organoleptic changes of the animals and some toxic effects in 1 week. And after using the concentration 500, 1,200, 4,000 milligram per kg body weight, we check the first 24 hours, the observation is done in a very minute way. Then next 72 hours, we carefully check whether the animals, the holistic approach is going to be changed or not. So we confirm that this extract is not acutely toxic indeed. And then we finalize to go for animal model test, I mean in vivo test. Please. As you know, that antidiarrheal model, there are a lot of models. In our cases, we used magnesium sulfate-induced diarrheal model, barium sulfate-induced model, charcoal castor oil-induced diarrheal model. And I think these are literally these are well-cited models, so I should not explain all these models. So through these models, we produce diarrhea, and how we calculate and how we confirm that animals are under diarrhea. Once we confirm that animals are diarrhea through the motility of the gut, their fecal content increases, then we check then some other parameters. For example, before going this model, we check this whether plant material are antimicrobial or not, because many of the diarrheal events are happen through a lot of microorganisms, and those microorganisms are truly claiming that whether this sort of plant materials are active against those. We use Salmonella dysentery, then Shigella dysentery, E. coli, these two microorganisms, because they are very much usual to cause the diarrhea. So we check their zone of inhibition, whether they are suitable or not. After checking the zone of inhibition, we found that the extract, they're good enough. They're good enough to control the microbial growth and their propagation. So ensuring that we went to castor oil model, magnesium sulfate model, and charcoal model. Let me clear and let me briefly describe my result. We see the first one. First one is the castor oil-induced diarrheal model. The percentages of inhibition, if you see in that case, we see almost all are very much closer to the reference drug loperamide. And that average number of fecal excretion, this is obviously lessened and reduced because of the extract administration. Similarly, in the other models, we can say the percentage of inhibition, we say that the extract in different doses all are effective, and this if average number of fecal excretion is also very much significant, statistically significant. So this led us to collect the serum of the blood from the animals and analysis through some biochemical analysis. So could you please go for next slide? Yes. So I have already tried to overview. You see the data. So we can go for the next slide, please. Yes. Here we explain actually the average number of fecal excretion and the percentage of inhibition of their fecal excretion. Next, please. Yes. So in this case, this is another magnesium sulfate induced. Also, we see that the percentage of inhibition is very much statistically significant, although we forgot to use the error bar in these cases. But average number of diarrheal fecals is also very good. And in this case, we say the medium dose, particularly the ethyl extract, is very much potential than the two other extracts, chloroform extract and methanol extract. Next slide, please, because we have to manage the time. We would like to finish in time. Yes. In this case, we see the castor oil-induced gastrointestinal motility test. You see, the motility in the case of the reference drug is 52, whereas in our case, the chloroform extract is 40. That means this is very much close to the reference dose. Whereas the peristalsis index is getting increased by the ethyl acetate extract is very well, very promising result. And in the case of next one, we see that the standard drug shows the 85% percentage of inhibition, and the extract one, we mean the chloroform extract, is showed 74. And according to the data of peristalsis index as well, it showed that the ethanol extract is showed the highest effect. So to some extent, the result varied among the extract to extract. However, the consistency is that the effect is close to the reference drug loperamide. Next one, please. Yes. So in the case, you see the barium sulfate induced model where we saw the intestinal gut lumen, and we measured the length of the gut lumen, and you see the extract decreased the whole length of the intestinal gut lumen. That is very much significant. And in barium sulfate induced gastrointestinal motility, we see the percentage of inhibition is much better than even reference drug. This is important. Reference drug showed 52% only, whereas methanol extract showed 55%. Similarly, peristalsis index is showed that chloroform extract is showed 71, and the reference drug showed 88% of the peristalsis index. So, which gives us, next slide, please. In this slide, we check the serum sodium level, potassium level, then serum chloride level, and serum IgE level. You know very well that in most of the diarrheal cases, the inflammatory bowel movement is happening, and in that case, the IgE, immunoglobulin E, is increased in much higher level. So this is a challenge whether we can manage the IgE level. Similarly, the sodium, potassium, and serum chloride levels are important, because once we lose the membrane gradient during diarrhea, in that case, we see that the sodium level outside the cell, it's usually we see higher, 120 millimole, and inside it's only 10 millimole, and potassium inside is 140 millimole, outside is 10 to 20 millimole, whereas chloride is 7 to 18 millimole per liter. So this is quite changed during the diarrheal cases. So in our case, we see that the sodium level in case of castrol induced and barium sulfate induced is significantly managed, and very significant is the IgE level. You see the castrol induced model and in the barium sulfate induced model, we see that the chloroform extract in the case of castrol induced model, in the case IgE level is not managed. However, to other sample, I mean chloroform ethyl acetate extract and methanol extract, they manage the IgE level in a very significant way.
Last 2 minutes.
Next, please. Yes, I'm going to finish. Yes. So next one, we collect the gut mRNA expression using the RT-PCR and 3 antioxidative enzymes, catalase, superoxide dismutase, and glutathione peroxidases. We can go. This is simply the primer we use, so we can go for next slide, please. And in this case, we see the plant extract. It was not able to upregulate the expression of glutathione peroxidase, but interestingly, catalase and superoxide dismutase expression was increased multifold through this administration of the plant extract. So this is very significant. And from that, we went for the GCMS analysis. And next, please, we'll go faster. Yeah, this is GCMS analysis. Next, please. And we targeted some plant compounds. We targeted some compounds, and as well, we went to go for the plant HPLC analysis, preparative HPLC analysis. Next, please. And from this analysis, we confirmed one compound, the oxoaporphine compound, and that compound, this is to some extent, this is the first time from this plant, although the compound is not new. However, this is from this plant, this is new. So using this, next please, the compounds, active compounds, we checked the binding efficiency and energy required to actually to make the ligand receptor interaction for well binding of the drug. And next please. Yes, and in that case we found that our docking analysis showed the R-oxoaporphin and S-oxoaporphin, they can increase the M3 muscarinic acetylcholine receptor binding and 5-hydroxytryptamine 3 receptor binding, and the binding energy was much negative. In the case we see that minus 5.324, which actually indicated that these compounds of Licia glutinosa, they are quite capable to make a good efficient binding with the ligand, the receptor of the drug susceptible to be bound for antidiarrheal activity. And we conclude that the oxidative homeostasis is very important. And in that case, not only gut, but also liver is also affected. And we find that since sodium oxyperoxide dismutase and catalase activity are getting upregulated, so they might have a very good role to protect the intestine and serum markers for optimizing the diarrhea. Next, please. So we are usually publishing in phytomedicine, biomedicine, pharmacotherapy, Virangana food science, Willy, then molecules, nutrients, biomedicine, BMC complement alternative medicine, Biomed Research International, and all others. We usually all are either Elsevier, Willy, then strangers and molecules. This is our prime sources to publish. This is my research. Next one, please. So this is my research group. Next page. This is my research group. I have 2 PhD students, 6 MPhil students, and 8 master's students. And next, please. This is our research laboratory. We have currently the collaboration with some other countries, Malaysia, Thailand, Saudi Arabia, and we are funded by our Ministry of Science and Technology and some other organizations. And we are welcoming all to make collaboration. Thank you so much. Thanks as well. I'm thanking the organizing committee again to invite us. Thank you so much for your patience here. Thanks. Questions are very much encouraged. Yes.
Thank you, Professor Atiyar Rahman. It was a great presentation. Is there any question? Is there any question? I think no.
Well, I have some questions.
You okay?
No question to him because I'm very interested about the Licia. So the Licia is many species around the world. Then why you choose this as the first stages for your investigations? Because of this is a kind of spices or Indian lore, as they say.
Yeah, thank you so much for your valuable question. Actually, if I tell the story, when I was in school, in that case, once my mother, she collected some plant leaves and she mashed it and just fed me the watery part of this plant leaves. And I was suffering from diarrhea. Then I saw that after 1 or 2 glasses of drink, I got quite fine. No drug, nothing. I asked my mother, what is this? Then she told, this is nothing. This is called Madapata. We mean our local language, Madapata. And they're very interesting. Once we came to study biochemistry and doing research in alternative medicine and functional food, then I again remembered that plant my mother introduced me to use in diarrhea. So that was the reason why I have chosen the plant for diarrhea. However, there are a lot of local uses, but we hardly found the scientific articles and publications in regarding this plant. So thank you so much.
Thank you, Jose. This is very good, warm-hearted story about your mother, right?
Yeah.
Is there any more question? I think no. Thank you very much again, Professor Atir Rahman. Hope to meet you the next time face to face. We are sorry this time we couldn't manage because of the governmental issues. But welcome to Turkey. I hope next time we will entertain you in Turkey again.
Yeah, maybe I will connect you for some other issues regarding
Yes.
conference certificate or something else.
Surely,
Okay, thank
surely.
you. Thank you so much.
Thank you. Takashi-san, you can attend. Professor Nazmi Arda.
Arda, okay. So now, next invited lectures is Dr. Nazir Arda. So his title is Honeybee Product and Their Relationship with Flora. So could you start it?
Nazlı Hocam.
Arda.
Nazlı Hocam.
Arda coming.
Nazlı
Please.
Hocam, where is she? Where is she? we should hemen Nazlı Hocam burada mısın? Hemen şeye devam edelim. Marina geldi mi? We can attend with Marina Spino. Marina, are you here?
Hello, everybody.
You can
you
start.
hear me? Can you see me?
Yes, great. Takashi-san, Marina, you know.
I know.
Yeah, you can introduce her.
Okay. How is your country right now? Coming the cold?
Not too good. Not too good. It's not too cold, but it's a lot of COVID.
I see, right. So I cannot say anything more. Anyway, so let's start your topics today, okay?
Please, can you guide me? How shall I share my screen, or I send my presentation to Nazim?
Okay. If you can send, or you can share middle of the
I sent
screen.
it to you, but I don't know how to share.
If you open in your computer now.
Can you see it?
The first, open your computer, your presentation.
Yes.
And then come to share screen, green.
Green color,
middle of the bottom of the screen.
Okay.
Share screen.
Okay. I see it now.
Yeah, share screen. Yes, coming.
Marina coming up. Oh, good.
Marina coming. Open your presentation.
Okay. It's open.
Yeah.
Yes.
Can you see it?
Already.
Yeah, okay.
Okay.
Right. Go ahead.
So just before starting, I wish to greet you all. I'm very happy to be with you again in this very, very nice symposium, which we are already attending for the, I think, 6th time. On behalf of my group of researchers, please allow me to send you greetings, to forward you greetings. I am very unhappy because of this situation that we couldn't attend in person, but I wish to thank Nazim and Sevgi, who are our friends already, for all the support and assisting us all along. And now I would like to start my presentation. How much I have, Nazim? How long can I talk? I cannot hear you, Nazim.
Yeah, 20. Professor Marina, you have 20 minutes.
20 minutes. Okay.
Yeah, 20
thank
minutes.
you very much.
Okay, thank you.
So this presentation is, in fact, a general presentation, but intends also to familiarize you with some of our results, long-lasting work for over 30 years on antimicrobial and immune-stimulating effects of plants in veterinary medicine. We are veterinarians, so we are a little bit outside of the group of plant scientists, chemists, but we work a lot on plants and their biological effects. You are familiarized with the relatively recent One Health, One Welfare concept, which connects humans and animals in the same health frame, and it stresses similarities between them as hosts for pathogens, also sharing their possibility to respond to extracts from plants which have biological effects. Phylogeny-depending structure, integrity, and responsiveness of the immune system is very, very important in preserving health of the host during microbial aggression. And a long history, drugs were sought in this field. So medicinal plants represented sometime the only resource for humans, and along with their therapeutic use in human diseases, they were used for treating animals. In some countries, these ethnobotanical resources represented the only accessible treatment, especially in small communities. And most of the plants were used for numerous effects. I'm going to go into more depth in a minute, but they were used to support welfare of animals and used as antimicrobials. This work compares the effects of different plant families containing various chemical compounds, and it's kind of a review of their immunological and antimicrobial effects. As I said, I'm going to support that with some illustration of our work. The plants with synergistic effects, which could have immune modulating and antimicrobial effects, especially efficient against multi-antibiotic resistant strains, would be an asset both for human medicine and also for animal medicine. If we could find these plants that would share multiple effects, they could be used also as adjuvants for veterinary vaccines. You know that protection against diseases is, if we grossly speak, ensured nowadays by vaccination. And another way of fighting diseases is antibiotic therapy. So if we could find plants which share antimicrobial and immune stimulating effects, we could further use them to enhance immunity and improve welfare in veterinary medicine as well as in human medicine. Such plants are ranked for their overall medicinal usefulness. I would start off with talking a little bit about the epidemiological triangle, which is, of course, very popular in medicine, both human and veterinary medicine, where you can see these three elements, these three corners of the triangle, which are the host, the agent, and the environment. Obviously, if there is no host, there is no disease. If there is no agent, there is no disease. But the interaction between these two occurs in a certain environment. And you can see indicated on this slide that each of these tips of the triangle has certain traits which are interactive, and they very strictly condition the development of the disease or preservation of health. As host is concerned, the phylogeny, the position of the individual on the phylogenetic scale, species, breed, age, their sex, their immunity above all, and also raising technology are very important in influencing the health status, the welfare status, and also susceptibility to diseases of the host. The agent, its biology. Is it a bacteria? Is it a virus? Is it a prion? The fitness to the host. Some agents are adapted to certain hosts. The activity of the environment, the pressure exerted on the agent by the environment. Antibiotic treatment is such kind of pressure from the environment. And this pressure exerted on the agent could lead to selection. So these are very important elements concerning the agent. The host can provide the environment for the agent to be able to multiply. On the other hand, environment shares some traits, temperature, humidity, altitude, latitude, which can influence both the host and the agent. You can compare, for example, intensive raising of animals which are crowded in a shelter where humans can control the environment, to other animals which are kept on the outside highly in the mountains, for example, where there is no control, just natural control on these temperature, humidity parameters. Then overcrowding of certain animal shelters, animal buildings or houses would lead to immune suppression on one hand, but also would enhance transmission of diseases. Pollution of the environment is one of the very important factors which can control or influence the agent. Some of the agents, like bacteria, are very well adapted to heavy metal polluted environments, and they become stronger and could attack the host more efficiently, being also resistant to heavy metal pollution. I think that these few words about the three elements which are involved in disease development gave you an idea about how important would be to be able to fight the agent's capacity to attack the host, controlling the host's resistance, but also the environment. This is a very scary statistic which I found on the internet. A forecast on the mortality, it's about people due to different causes. And you see by 2050, antimicrobial resistant infections will be in the top, causing millions of deaths all over the world unless we come up with some solutions. Just 2 words about antimicrobial resistance, because it's one of the topics I'm going to tackle as solved by use of medicinal plants. This is an overall image of the bacteria, how they can fight antibiotics, and you have listed some of the mechanisms. We're not going to go into detail, just you should remember that the microbes have in their genome or extranuclear resistant genes. They harbor these genes. Actually, at this point, microbiologists consider that there is no microbial pollution of the environment, but it's the plasmidome, the cumulative, the sum up of the plasmids which ensure for the bacteria antimicrobial resistance. Those are the real pollutants. Then another mechanism would be the possession of antibiotic degrading enzymes or antibiotic damaging enzymes of the bacteria. And the fourth one is the efflux pump. Just some of the mechanisms. They are incredibly adaptable to the environment, adaptable to antibiotics, and this would be an explanation why the antibiotic resistance spreads so quickly. And this is one of the schemes which highlights, of course, everybody's familiarized with intra-hospital infections. Everybody knows that some people hospitalized for different reasons, like surgical reasons, can come out from the hospital with different infections. This site is that one I'm going to stress. The role of animals. Of course, animals are treated with antibiotics. They will shed antibiotic residues. Further, they will accommodate, in brackets, antibiotic residues in their products, like eggs, like milk, like meat and meat products. Therefore, gradually coming in contact with these residues, people will acquire antibiotic resistance. Another way to share antibiotic resistance between humans and animals is just their close contact. Caretakers will share their bacterium with the animal bacterium they take care of, and this way between these two types of hosts, there will be a transfer of plasmids which can further increase antibiotic resistance. The environment, as I mentioned before, the vegetal farm, which eventually provides feed for animals, can also further increase this antibiotic resistance by accommodating bacteria which are non-pathogenic themselves, but they share these antibiotic resistance plasmids. Nobody knows where these came from in these non-pathogenic bacteria, which never encountered any antibiotics. Now, of course, I mentioned something about feed. The influence of feed on the host is obvious. If there is no food, there is no host, because it's supportive of all the energy needed by the host to develop and keep its welfare and health. And of course, one of the important aspects of the host which enhances this survival is the immune system. And the pathogens which will attack the host will first encounter the immunological elements of the host, which recognize them as non-self. This is a very complicated slide. Don't worry, I'm not going to go through it all. It's just an explanation of the very complex answers which a pathogen, and you see on the top some pathogens listed, including bacteria, viruses, fungi, and also parasites, which attacking the host will trigger various responses. First, the non-specific responses, and then the adaptive specific responses, which end up by killing bacteria, viruses, protozoa, or other aggressors. So the pathogens are in the middle of the immune response, and they can launch both humoral responses, as from the adaptive part of the immune response, and cellular immune responses or cell-mediated immune responses, which finally result in antibody synthesis. Which are the potential effects of plants on animals? And you see listed many. First, obviously, on the digestive system. They will, well, containing fiber, will enhance digestion, but also containing compounds which will enhance digestion of different proteins they ingest, or will increase the availability of vitamins from the food, for example, improve mineral absorption, and this way they increase food use. Due to their antioxidant capacity, the ingested medicinal plants as such will fight the pathogenic bacteria in the digestive tract, and this is another mechanism. Oh, they are very complex, and there are many ways medicinal plants ingested as such could fight disease in the digestive system. Then again, due to their compounds, and we'll go into this complicated structure of medicinal plants in a minute, just very briefly, they have this antimicrobial activity, and you will see that various compounds from plants can fight even multi-drug resistant bacteria. And of course, the central piece in this overall picture would be the enhancement of the immune system, ending up with increased production of immune globulins, increased numbers of white blood cells, increased antibody production, higher antibody levels, and then the overall stimulation and enhancement of the immune response. All these combined will lead to improved productive and reproductive performances of animals, which we aim at raising them on farms. The effect of medicinal plants are very numerous, and just to list a few of them, anti-inflammatory effects, antimicrobial, which I already mentioned, pain relief effects, analgesic effect, and antioxidant effects, which I also indicated before. What is interesting here is that there are very numerous plants which were tested for their anti-inflammatory effects, well, derived from human medicine. Then antimicrobial effects are numerous plants, but less here listed. And as we go to antioxidant levels, they are less and less listed. That's not a realistic picture, because plants are tested for multiple effects, not only for particular effects, but also it's very interesting to see that they share multiple effects. And I marked in the same color here plants which are listed under different effects. Anti-inflammatory, for example, chamomilla, will be also antimicrobial and will be also antioxidant. part of its antimicrobial effect is based on its antioxidant activity. Similarly, Echinacea is an anti-inflammatory plant with antimicrobial effect, and it's also very well known for its immune stimulating effects. In this table, which I took from bibliography, and you see indicated in the left upper corner the authors of this table, the very worrying part is all these marked in red, because there are numerous microbes which were tested for their pathogenicity or known for their pathogenicity, such as multi-drug resistant E. coli. This is MDR, multi-drug resistant. That means that these strains are very resistant to numerous antibiotics, and sometimes to all those which are tested. So what I marked here are all multi-drug resistant strains, and opposite in blue, you see how many weapons, would I say, we have provided by plants to fight this kind of multi-drug resistant bacteria, like terpenoids, flavonoids, saponins, tannins, polyphenols, organic sulfur compounds like in garlic, cinnamaldehyde from cinnamonum, flavones in multiple positions on this table from different plants. There are many plants that share some compounds like essential oils, for example, which are very efficient against multi-drug resistant bacteria. All these pathogens you see listed here, Klebsiella pneumoniae, E. coli, Staphylococcus aureus, are also human and animal pathogens. So they can be found in people and also in animals. So this is a fortune, would I say, provided by plants at our hand to fight against multi-drug resistant bacteria. Now, of course, because we usually normally do not use plants as such, but we use them as extractions, I just listed from a paper I read on the pros and cons of extraction methods, and the overall conclusion is that the evaluation and selection of pre-extraction preparations and extraction methods are depending on the study objectives, samples, and targeted compounds. That's very important. How can we assess the immunological effects? There are various methods. I mentioned already which mechanisms are involved, and also we have to tailor the extraction methods and the plant compounds we use in testing biological effects to the aim we look at. So immunological testing can be done either looking at the number of immune competent cells when we administer extracts in vivo, or we can look at in vitro effects of various extracts obtained from plants. With essential oils is a little bit of a problem because they are oils, and all these tests, like immunological tests, go on in watery fractions. So sometimes emulsifying compounds need to be used. The antimicrobial testing is, some of you are familiarized with. We have this diffusion method, which you see here, indicating the more enhanced effect or no effect of some of the plant extracts. Of course, against various bacteria, we want to see if they are efficient or not against. Another method, and
Marina, please. Please,
yes, shall I finish?
Yeah, please.
Okay, so then I'm going to just very quickly go. It's results of our previously published studies. We worked on numerous species, and you see these are all published or communicated. Again, leading to the conclusion that the effects of plants depend very much on the animal species and depend very much on the plant species. So we worked on fish, we worked on equine, we worked on chickens, we worked on bovine. The conclusion of this study is that the investigation of biological effects of plants in veterinary medicine is far from being closed. the antimicrobial and immunological activity of different plants as facets of the same complex healing potential of plants, they could offer need further in-depth interdisciplinary studies. I just want to very quickly present the very young group that worked a lot on this kind of tests, and our lab assistant who provided all the materials, and thank you for your attention.
Thank you for your presentation. So any scientists ask the how to the special topic something. Please, hand up.
I want to stop sharing my screen, and I don't know how to do it.
It's okay. It's okay. Don't
Yeah?
touch that.
Okay.
Yeah, don't
Okay.
touch after the finish.
Yeah. Okay. Okay.
So you are going to the study about the COVID-19 something right now?
Sorry? No, we don't have biosafety level to do that. We can't do plants, but some people did, as far as I know.
Okay, because plant research can help the whole future of our human being, right? So
yes, that would be interesting. Yes.
Interesting. Anybody ask the some?
If I can ask a question, Srendo Farouk, most of people who look in plant extracts and for immune modulation look at compounds that suppress immune response, that are kind of anti-inflammatory. So, but that of course would make you more vulnerable to bacteria. So am I right that you should be looking for pro-inflammatory compounds in your plants if you want to boost the immune system?
Well, not necessarily, because inflammation is or could be the first stage of an immune response, but we could target specific cells, for example, T lymphocytes, B lymphocytes, which are part of the adaptive immune response, and they are members of the antibody synthesis process. And therefore, by triggering their response or improving their responsiveness, we can achieve a higher level of antibodies, which will kill bacteria and improve the health and welfare of the animals. And also, we will obtain, probably, we didn't look into that, memory cells, which will keep their memory for the antigen, obviously. This is why I mentioned at one point that plant adjuvants will be very handy if we would have them, because if they are themselves antibacterial, they are immune enhancers, then they would have a double effect. They kill the bacteria and they are antimicrobials, that's proven, and then they will enhance these cells to respond to the bacteria, and they indirectly, facilitating the antibody synthesis, will come back to the bacteria and kill them both ways, so directly and indirectly through the antibodies. So not necessarily pro-inflammatory, because that will be pain, you know. Okay, we have a problem with essential oils and extracts which contain alcohol, because you can't just inject them. They will trigger a pro-inflammatory response. So we still need to find a way to dilute them to a concentration where they are not harmful injected, for example, locally. This is why orally is more favored administration route at this point. Does it respond to your question?
Yes, thank you very much.
Okay, thank you for the question.
Maybe I already asked.
I can. Thank you, dear Marina. I want to introduce and invite Professor Dr. Nazlı Arda from Turkey. Nazlı Hocam.
Thank you very much once again.
Thank you. Greetings to Romania from here. Nazlı Hocam.
Okay, Marina.
I think there is some problem for Professor Nazlı. We should call her. Sorry. 1 minute. Yeah, sorry. Tamam.
I'm here. I'm here. I'm sorry.
Welcome, welcome.
Don't we have any break? Coffee break?
So we are sorry. This time everybody give. We don't have any coffee break, so we have some discussion, and we couldn't give coffee break. We serve you.
Okay.
Next time, coffee.
Okay. Let me give some minutes to open my presentation. Okay. Sorry.
Okay. Takashi-san, it's in the night in Japan, huh? Dinner time.
So now,
do you have Turkish tea, chai?
Thai huh?
İnce chai.
I like the
İnce belli.
Yeah, Germany. All the time I take the Turkish tea.
After this, Japanese professor prefer Turkish tea, chai in the special glass, Turkish glass. İnce belli chai.
İnce belli chai. İnce belli. Yeah.
So we are very happy to entertain people. Hocam hazır olunca tamam.
Yes, I am ready. I think I'm ready.
Tamam.
I'm sharing my presentation with you.
Okay. Tamamdır hocam. F5 yaparsan. Okay. Tamam.
Okay, dear chair, I'm very sorry to be late.
I have a question.
Dear chair, dear all members of organizing committee of MESMAP 6 and dear participants, first of all, I would like to thank Professor NAZIM SEKEROGLU and Associate Professor Sevgi Gezici for their kind invitation. I'm very happy to be here to meet with you, even though online. So let me introduce myself first. My name is Nazlı Arda. I work in Istanbul University as a full professor at the Department of Molecular Biology and Genetics, and as a director of a center on biotechnology and genetic engineering. Actually, I have been working on medicinal plants for years since my PhD degree. But recently, my scientific journey is proceeding on various biological activities, including antioxidant, antimicrobial, antiviral, anti-cancer, and anti-inflammatory of natural products, but mainly honeybee products and their metabolites and their action mechanism on molecular level. Today, I'll talk about some details on strong connection between the plants and honeybees and honeybee products, of course, and share some ideas on this topic along with some recent studies on this topic. Since the immemorial time, both plants and bees have coexisted on Earth, we know that. And we all know that this relationship between bees and plants is a symbiotic type, because plants actually, plants serve as food and shelter for bees, and bees help plant reproduction. And of course, biodiversity of plant species. Nectar provides carbohydrates, and pollens provides proteins, mainly food constituents of honeybees. Some bees also need shelter provided by plants for them. In turn, bees help plant reproduction, and they cross-pollinate flowers, diversify the genetic background of seed, and help plant species reproduce and survive. Diversity among species, including agricultural crops, medicinal and aromatic plants, rely on bee pollination to some extent. It is predicted that nearly 16% of world's flowering plant species are pollinated by bees. All living things, mainly human beings, take advantage of this mutual interaction by different ways. In point of plants, first of all, plants are primary source of energy in nature and are unique and God-gifted source for human beings, mainly as food. As you all know, they are also marvelous sources for medicinal and aromatic compounds. In addition, some chemicals, fibers, such as cotton and linen, biofuel, and construction materials. So timbers, sorry. They are also very important for economical and ecological situations. For ecological balance in terms of water, land, and air qualities and climate, briefly, geographic structure and characteristics of a region. Finally, they have great economical and social impacts by various activities such as agriculture, forestry, pharmaceutical, chemical, food, textile, cosmetic industries, tourism, and urbanization. So, on the other hand, bees are a key player in the human food ecosystem. They pollinate more than 1/3 of all the plant foods. It means without bees, there is no food in our world. Apart from their roles in pollination and biodiversity, bees also serve mankind by offering or sharing their products with high nutritional value and beneficial properties. Because of close interaction between plants and bees, both are indispensable for health and wellness of people, of course, of all animals, all living things, and for sustainable economy. There are around 20,000 bee species worldwide defined to date, and approximately 2% of these species are honeybees and bumblebees. The other 98% are mostly solitary bees. Solitary means, you know, they live on their own, not in colonies with a queen and workers like honeybees and bumblebees. There are at least 9 honeybee species in Apis genus. Apis mellifera is the particularly well known as a honey producer, but also as an important pollinator of many crops. Although it is referred to as honeybee, there are at least 8. I say at least because some new species are known today, I don't know. And there are at least 8 other honeybee species in the Apis genus, such as Apis florea, Apis dorsata, serrana, etc. You can visit NCBI taxonomy webpage to see all the species of honeybee. Honeybees require habitats with plenty of flowering plants. They can live in natural habitats, agricultural areas, and urban parks and gardens. Their preferred habitats include orchards, meadows, gardens, woodlands, and virtually anywhere with plenty of flowers growing. Apart from their native populations, honeybees, especially Apis mellifera species, are managed by beekeepers. They are social insects, social bees. It means they live in a colony which consists of highly organized relationships between the members. Every honeybee in a hive exists to perform specific duties determined by her or his gender and age. Okay, who lives in a colony? The vast majority of a hive consists of worker bees who build honeycombs and collect pollen and nectar. These are the bees who visit flowers and are important in pollination. The queen bee mates with a drone and stores the sperm for long-term use, and she will continually lay eggs as needed throughout her life. She lays each egg in a box honeycomb, which is built by the young workers, namely nurse bees. And these bees, nurse bees, nurture and feed bee larvae. They take on a job of processing incoming nectar, feeding the queen, as well as making and capping the honey. A day in the life of a worker bee cover many steps. Foraging worker bees fly from their hive, sometimes miles away, and go some other flowers, from flowers to another blooming vegetation. Once enough nectar is collected, the bees bring it back to the hive, and the nectar is chewed up and deposited into honeycomb wax cells. The honey is still wet at this state, and newly hatched worker bees vigorously fan their wings over the nectar, causing the liquid inside the cells to slowly dehydrate. Drying process, along with natural enzymes added to the nectar, transforms the relatively thin liquid into a much thicker honey. So there is a unique process on this step, as you see. Once the honey is ready, the bees cap and seal the cell to keep it clean. There are more than 300 varieties of flowers and blooms that honeybees can visit, which means the taste, the color, and the smell will vary from honey to honey. What are the main components of the diet of honeybees? Of course, nectar for carbohydrates and water, carbohydrates rich in secretions, which is called honeydew, pollen for proteins, lipids, vitamins, and minerals, and water, especially compound-rich, dirty water for their essential minerals. Nutrition in honeybees can split into three phases. for colony, for individuals, and for broods. Okay, main honeybee products are honey, bee pollen, royal jelly, propolis, bee wax, bee bread, and bee venom. Actually, all these products are originally made by bees for themselves, mainly for nutrition and protection of the hive members. Honey is produced by bees collecting nectar, and honeydew for use as sugar consumed to support metabolism of muscle activity during foraging or to be stored as a long-term food supply. Bee pollen is used as food after processing that we mentioned before. And royal jelly is a honey bee secretion that is used for nutrition of larvae and adult queens. And the worker nurse bee secretes royal jelly from its spastic glands, and this is fed to the larva of queen, worker, and drone bees. Propolis, by the way, or bee glue, the other name is propolis, is created from resins, balsams, and tree saps. It is consumed by humans as a health supplement in various ways, and also used in some cosmetics, you know. And bee wax is secreted by worker bees, and they use this product to form the walls and caps of the honeycomb. And bee bread is essentially pollen collected by bees and packed into the bee brood cells, mixed with bee digestive fluids and nectar. The bees then seal these cells with honey and store in hive for later consumption. Bee venom, by the way, it is out of our range, but it is used as defense mechanism of the worker bee to protect itself or its colony. Honeybee products have beneficial effects on human health and possess several medicinal activities. First 5 are commonly used by humans because of their nutritional and medicinal traits. They have many medicinal activities such as antioxidant, antimicrobial, antiviral, anti-cancer, anti-inflammatory, etc. For example, humans have used honey for medicinal purposes for thousands of years. It is also used, widely used in traditional Chinese medicine and Ayurveda, you know, and some of the major documented uses were wounds, burns, asepsis, ailments such as cough, insomnia, and gastrointestinal disorders. Bee bread, a diet for bee larvae and young bees, appears in the market as a functional food more recently, and the queen bees and their larvae are also marketable. Bee venom, also known as apitoxin, is a colorless liquid that can lead to localized inflammation or extreme cases, such as severe allergic reactions. But it has been used as an alternative therapy called apitherapy. It is given as a shot for rheumatoid arthritis, nerve pain, multiple sclerosis. Okay, what is the content of bee pollen? Actually, all bee products, all honeybee products, contain various useful and bioactive compounds arising from plant sources. Those are visited by bees. For instance, bee pollen It is agglutinated form of flower pollens made by worker honeybees using nectar and salivary secretions, contain carbohydrates, fruit fibers, proteins, lipids, basic amino acids, as well as very active components and vitamins such as carotenoids, phenolics, flavonoids, sterols, terpenes, vitamins, and minerals. Most of the honeybee products can be used in the original state in which they are produced by the bees. However, some do not have a large market until they are processed into a suitable form for consumers to buy or are added to other ingredients to make new value-added secondary products. So because of the quality and sometimes almost mystical reputation of most primary bee products, their combination with other products, especially other bee products and plant secondary metabolites, usually enhance the value or quality of new products, these value-added products. And the combination of several bee products or addition of a plant-derived ingredients synergistically increases their beneficial significance beyond their individual biological values. So, thus there are thousands of commercial honeybee products in the market, as you see in this figure. Composition and quality of both diet of honeybees and their attracting commercial products mainly depend on the floral and water resources, geographic origin, climate, and soil. Besides, honeybees alter their foods, thereby quality of their products according to the season. Both resources and metabolic processes demand this alteration. For example, phenolic and flavonoid contents and the antioxidant activity of autumn propolis are higher than those of spring propolis. And finally, beekeeping activities to some extent, like handling, transportation, and storage conditions, change their compositions and quality. Unfortunately, the most pressing threats to long-term bee survival include climate change, habitat loss and fragmentation, invasive plants and bees, low genetic diversity, and pathogen sprayed by commercially managed bees and pesticides. Decline or disappear, in other words, colony collapse disorder of honeybees arising from a combination of these factors is also a serious threat. Consequently, beekeeping sector, together with the whole ecosystem and agricultural sector depending on pollinators, suffer from these harmful processes. There are several efforts by non-governmental organizations, by governments and governmental unions to reduce these threats, to prevent honeybees, to make their products safer, and to improve biodiversity. For example, the World Bee Project is a British social enterprise, the first private organization to launch a global honeybee monitoring initiative to inform and implement actions to improve pollinator habitats, create more sustainable ecosystems, and improve food security and nutrition by establishing a globally coordinated monitoring program for honeybees and eventually for key pollinator groups. It is the UK equivalent of a US benefit corporation known as B Corp. There are some other efforts to improve biodiversity, especially in European Union, such as 2020 Biodiversity Strategy, which introduced in 1910, and it covers many attempts, including EU pollinators initiative, works on decline of pollinators. And same strategy still going on with a new version, 2030, the biodiversity strategy. And for the next 10 years, European Union will focus on a new European-wide network of protected areas on land and at sea, concrete commitments to restore degraded system on a global level.
Nazlı hocam yavaş yavaş.
Okay. Another international efforts called Apimondia. They exist to promote scientific, technical, ecological, social, and economic apicultural development in all countries. Turkey is considered to be one of the countries richest in plant biodiversity in the world, as you know, and our country is home to more than 12,000 nature plant species, which about 450 among are attractive for honeybees. And we have 5 species, honeybee species. So we focus on these bee products, honeybee products, especially for their several biological activities and understanding of the action mechanisms. Here you see our ongoing research on honeybee products. All of them are graduate projects, such as effects of various extraction and fermentation processes on the antioxidant capacity of honey, propolis, and pollen samples, bee bread production via pollen fermentation using various probiotic strains, etc. You will see all the names. And we have some studies on biotechnological production of a bioactive royal jelly peptides or some biotechnological approach in cosmetical product development from royal jelly. We use several methods, conventional methods, of course, and some living systems, bacterial yeast and animal cell cultures, or collected or commercial materials in our assays, as well as some molecular techniques, for example, metagenomics, to identify microorganisms involved in these processes. This figure represents a work conducted for monitoring the pathogens in a bee population and identifying microorganisms that improve bee health and give some information on how honeybees process their foods using microorganisms and identifying preferred flora species in landscape. A part of our studies will be alive in the future, so I would like to show this figure from another reference. Thank you so much for your kind interest. I wish to get rid of COVID-19 and the coronavirus as soon as possible, and to meet in the next meeting safely and work arm in arm as before. Thank you.
Takashi hocam.
Okay. Somebody question to the bee specialist. I have a question anyway. So we have some project in Tokyo about the bee, because of our bee is a little bit small type of the bee. So my question is, I heard in the travel zone of Turkey, nearby the South Black Sea, they are very nice, bee, product. Then I heard that some group B only collect nectar from the one type of flower or not. Is it true or not? Because the bee have some behavior of the one plant, they like it or not.
Sorry, I lost my video again. Where
I mean,
are you? Where are you? I can't see. You see me? Are you seeing me?
okay. Can I show you the one question to the sharing the my, just to show my question here.
I can't find.
Maybe. Okay.
I don't understand what's happening now.
Could you see my question here?
No, I lost my. Maybe I will find. I will find it. Sorry. I'm not very familiar with this. Okay.
You can see my.
Could you please repeat your question?
Hocam görüyor musun ekranda?
Okay.
I görmüyorum.
I have a question. So,
Hocam kendisi paylaşıyor şu anda.
The bees are.
Please share your question, Takashi-san. Again.
What?
You can share your question. You can share.
Okay, I can show again. Could you see my question on the wildfire?
I have heard that the same group of bees only collects nectar from only one type of flower. Is that true? No, I think, as far as I know, it's not true. It's not true. But I know worker bees lead the other bees in the same colony, of course, to visit better flowers when they find a better one. Do you understand me?
I understand. So second question, the bee used to tell the mate about the today's nectar sources. If the nectar is very fantastic, they like it. So dancing in the behind the shaking the hips.
Yes, I agree with this preference of the bees for the flowers which their nectar content is high. Yes, I agree with the second one.
So your Turkish bee is a Western type of bee, is mainly or not? What kind of bee in your country?
Maybe you missed it. We mostly have Apis mellifera, but different species, subspecies of Apis mellifera.
Thank you. So we have no time. Maybe final. Maybe.
Thank you.
I think
so. It's time in Japan to dinner, in Turkey lunch, in Western part breakfast. I think. Teşekkür ederim.
Yes, we would like to thank you all our participants, invited speakers, and now we will begin in the afternoon at 2 p.m. and we are going with the invited speaker sessions. Please be online at least 10 minutes before the session. Thanks again for your valuable contributions and valuable presentations.
See you afternoon. Thank you, Professor Randolph. Thank you very much, and Takashi-san. And we are sorry we couldn't arrange the time. And afternoon you can sleep, Professor Randolph. Your microphone.
Yes, yes, it's a pity, but yeah, I will go to Izmir one day in my life.
Yeah, but hope to see you next year in Turkey. We
Yes.
will entertain you. Thank you. Thank you for
I'll be
coming.
very happy to.
Thank you. Bon appetit. Have a nice dinner. Enjoy your breakfast. See you the next session afternoon. Bye bye.